Check, check, check. 1, 2, 3, 1, 2, 3. Hello, hello, 1, 2, 3. Check 1, 2. Check 1, 2, 2. 1, 2, 3. Check 1, 2. 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 1, 2. Check, check, check 1, 2. Check 1, 2. Check, 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 check. Check 1, 2. Check, check, check 1, 2. Check, 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 check. Check 1, 2. Check, check, check 1, 2. Check. Check 1, 2. Check, check, check 1, 2. 1, 2. Check, check, check. Check 1, 2. 1, 2. Check, check, check 1, 2. Check, 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 check. Check 1, 2, 2, 2, 2, 2. Check, 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 check 1, 2, 2. 2, 2, 1, 2, 2, 2, 2, 2, 2, 2. Check, 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 check.
A very warm good morning to one and all. So we'll be commencing our program by 9:30. Kindly be seated. Thank you.
Hello. Okay. Good morning all. Institute of Advanced Virology is extending you a very warm welcome to all of you to the two-day hands-on workshop on monoclonal antibodies, an emerging era in biologicals, principles of production, applications in immunodiagnostics and therapeutics, to be conducted on the 8th and 9th of December 2022. To begin the inaugural session, I would like to invite the Administrative Officer of IAV, Mr. Manoj Kumar, to deliver the welcome address. Over to you, sir.
So, good morning all. A warm welcome to the workshop being organized by 2020. We are generously supported by Government of Kerala. So, our funding is from the Government of Kerala. So, unlike other research organizations and education institutions, so normally what happens, institution starts and then we think about creating the facilities. So, we were fortunate to have the physical infrastructure, whatever we are seeing here today, and that other buildings, it was ready built for us. So, we just came here, occupy the building and start making the research infrastructure. So, for the last 1 year, so we are also very quick to make the facilities here. In the last 1 year, we made all the research facilities to carry out the research in various domains of virology. So currently we are having 5 departments, and soon we will be adding 3 more departments in various areas of virology. So since the government is supporting us very much, it's time for us to reciprocate by giving results. You all know that Kerala has witnessed a series of virus outbreaks in the last few years, and we are working on various aspects how we can counter that. We already started few flagship programs wherein we will be delivering products which will help the state to overcome the difficulties due to these virus infections. And this workshop is also one of the steps in this regard, how we can increase our capabilities, build our facilities. So its workshop is also focusing on that part. So let me come to my duty to welcome all of you to this workshop. First and foremost, I will welcome the participants who are coming from various parts, various institutions from India to take part in this workshop. So a very warm welcome to all the participants. So I also welcome the distinguished scientists and academicians who, despite of their busy schedule, agreed to come here and provide their valuable inputs, hands-on training to the participants, etc. So I would like to welcome first our director, Dr. E. Srikumar, Director, Institute of Advanced Virology. So next, our first session will be taken by Padma Shri Dr. Lalit Kumar. He is the chairperson of Oncology and BMD Department, Artemis Hospital, Gurgaon. He will be attending the session online. So I welcome Dr. Padma Shri Dr. Lalit Kumar also. So next I welcome Dr. Devinder Segal, Staff Scientist, National Institute of Immunology, New Delhi. Welcome. Next is Professor Deepak T. Nair, Regional Center for Biotechnology, Faridabad. So, welcome to Dr. V. Gopalakrishnan, former adjunct professor of IIT Chennai. So I welcome Dr. Abdul Jalil, Scientist G. He's from Regional Rajiv Gandhi Center of Biotechnology, Trivandrum. So I welcome Dr. Chitra Ravind, lawyer and IPR expert. She's from New Delhi. So I welcome Dr. Tripti Srivastava, principal scientist from THSTA. So she will be joining us soon. So I welcome her also. So I welcome Dr. Chandresh Sharma, assistant professor, CSIM University also. He will also be joining soon. So we'll be having Dr. Santosh Kumar from RGCB in the afternoon session. I welcome him also formally. So I also welcome the industry representatives and other representatives of official sponsors who are generously supporting us for conducting this workshop. So a warm welcome to all the participants from the industry and the official sponsors. So I also welcome my fellow mates, all the EAVians who have been working relentlessly over the last few days to make this workshop a big success. You have to give a clap to them. So I'm sure that all of you will be going back with improved knowledge and new ideas in these areas of monoclonal antibody after this workshop. Once again, a warm welcome to one and all. Thank you.
Thank you, sir. To deliver the presidential address, I would like to invite the Director, Institute of Advanced Virology, Dr. E. Srikumar, to the dais.
Good morning. A very warm welcome to the Institute of Advanced Virology. So today you are sitting in a place which was in the minds of the people of Kerala, in the minds of the administrators of Kerala, maybe some 20 years back, like in 1998, they wanted to have a virology institute in the state. Even while we call Kerala as the God's own country, on the other side, we are also virus' own country. Thanks to the biodiversity of the Western Ghats, thanks to the vegetation, thanks to the climate, we get a lot of viral infections, and we had, and also our public health surveillance is very strong, and we detect most of these infections very early, and we were first in detecting the COVID, first in detecting Zika, maybe detecting many other viruses, and we also had outbreaks of new infections like Nipah, which was detected, and we also have a major diaspora traveling abroad and coming back, bringing more infections. So the necessity of having a virology institute was felt from a long time back. And this institute, the work of this institute, maybe the brain work of this institute started in 2017. The building was constructed immediately in 2018. We had a prefabricated building to begin with, like some 30,000 square feet, where we have the functional laboratories now. You will see that building on the extreme end. Then this is the new building that will be handed over to us very soon. So we'll be adding up another 80,000 square feet, and we'll be expanding our laboratories. And this building was inaugurated, our current building was inaugurated in 2019, and it became functional last year. And I joined here as the director 1 year back in the last October. And now we are proud to say that we have started 6 laboratories functional. We have a molecular diagnostic facility which can detect around 88 viral parameters, and we have tie-up with the hospitals in and around Trivandrum and also in and around the vicinity. Some 50 primary health centers are giving us the samples. So we are offering public health services that way, and we are doing outbreak investigation. So we are becoming fully functional. And to extend our activities further, we wanted to initiate a conference, a seminar or a workshop. Then there this question came up. So what should be our topic for the workshop? Naturally, any new institute with a virology, focus on virology, definitely we would have started with the infectious diseases, flavivirus infection, maybe respiratory virus infection. SARS corona was at its peak. We could have done with coronavirus infections, emerging infections. But we chose a very different topic that is called monoclonal antibodies. We were sure that this will get an overwhelming response from the participants, because the last COVID pandemic, one of the major interventions that we reinvented or rediscovered was the plasma therapy. And it started with the convalescent plasma therapy, then it went to monoclonal antibodies very rapidly. Thanks to the new technologies, the monoclonal antibody has matured enough to deliver very fast. We had the hybridoma technology in the beginning, and we had subsequently different humanized antibodies, and now came the recombinant antibody technology, which can make more or less antibodies with very less reactivity in the human system. So I am extremely happy to have a very overwhelming response from all of you when we advertised for the program. Maybe from the very next week onwards, we started getting applications and requests, and maybe at the end we had to say that now no more slots are available for the workshop, but maybe you are welcome to attend the talk. So we are streaming this live in YouTube, so I'm sure that there will be many more people who will be listening to the talk. And while choosing the faculty also, we were very careful. We wanted to have experts in the field who have hands-on experience and real background knowledge in the subject, rather than any book-based knowledge, but they had actual practical, strong and knowledge, and also theoretical background. So we are fortunate to have wonderful faculty members for the workshop, and I'm extremely thankful that they could find time to come over to Trivandrum and attend our program and give you enough exposure in the field. And actually, this response is a stimuli for us. We wanted to expose our institute to the people, to the student, and we want to build human resource setup. And I'm hopeful that the 2-day workshop will be extremely useful to you. And this antibody-based therapy, even though it was a classical approach long time back when Emil von Behring started the plasma therapy, he got the Nobel Prize for discovering the plasma therapy and antibodies. It has matured enough to a precision science now. And I'm very happy that all of you are here, and I wish all the success for today's program, and I hope that this will be extremely enlightening for you. And thank you once again for coming here and joining the workshop and making it a big success. Thank you once again.
Thank you, sir. Now I invite Dr. Rajesh Kumar Kalra, the convener of the workshop, to give an outline to the 2 days workshop.
So a very good morning to all of you. So our administrative officer Mr. Manoj Kumar and our director already have given a lot of welcome to all of you. So I will directly start with the business. So I will brief about what we are going to do for these 2 days, 8th and 9th. So we have to enable the capability of the young scientists, young students who are doing their bachelor degree, master degree, and even the PhD degree. And also we have invited here the startup companies who are trying to develop a ecosystem of monoclonal antibody-based diagnostics and the therapeutics. So in the 2 days workshop, we will be mainly discuss about the monoclonal antibody development, what are their different methods are there, and what are their different applications are there. And so we have a distinct speaker all over the India, and first speaker will be Padam Shri Dr. Lalit Kumar. Actually the first things come when we make any product, what is the real scenario use of that monoclonal antibody or that product. So immunotherapy or monoclonal antibodies are most commonly used in the field of the cancer therapy. So with us we have Dr. Lalit Kumar, who is treating the patients in the clinic for I think more than 20 years who are suffering from the cancer, and he was the head of department of the medical oncology, All India Institute of Medical Sciences, New Delhi. So he has worked with different clinical trials starting from phase 1, phase 2, phase 3 and even the phase 4 trials. So he has a lot of experience in that. He has a real scenario experience with treating the patient with these monoclonal antibodies. So we will have first our keynote address by Professor Lalit Kumar, and second is Dr. Devendra Kumar Sehgal, who is expert in the field of monoclonal antibodies, and still I am also learning from him how we can establish and learn more in that field. So he is also working in the field of more than 20 years in the field of immunology, and he will tell us what are the basic, how the development of B cell start, how these antibodies are rearranged, and how these isotyping switches happen. So he will be giving a detailed overview of that. And then we have Professor Deepak T. Nair with us. So one of the major aspects of the monoclonal antibody is that where these antibodies are binding. So we definitely need to know the place, that in the scientific term we call that we want to know the epitope of these antibodies. Once we know the epitope of these antibodies, we can also engineer these antibodies. So he has immense experience in the field of structural based designing of the monoclonal antibodies. And I really introduce that he has also been awarded with the Shanti Shauru Bhatnagar Award that equals to the Nobel Prize of the India. So he will be the best person who really tell us about how these antibody antigen complexes are made and how this information can be used for engineering of the monoclonal antibodies. So along with we have Dr. Tripti Shrivastava. So one of the real questions that comes in our mind is that we have to find out the target, against which target we want to make the monoclonal antibodies. So Dr. Tripti will be mainly focused on how we can design these antigenic bait in the different expression systems, starting from the bacteria, mammalian expression system, baculo expression system. So all those things he will be highlighting in his talk. So next we have Dr. Mohan, who is the senior principal scientist in IIB. So he will be mainly focused on the what is the role and application of these monoclonal antibodies in the viral infections. So till date, most of these monoclonal antibodies are used in the cancer therapy and the autoimmune disease. So from last few years, there are few monoclonal antibodies that are approved by the FDA for the use for the viral infection. So he will highlight more on that, what are the targets in the viral that we can use for making and developing the monoclonal antibody. So, and I will also be talking about what are the different technology platforms by which we can make the monoclonal antibodies, what are their advantages, what are their limitations, and how we have to select for which target, which method is the right method for making the monoclonal antibody. There are different technology platforms, but we have to select which method is right for us for making these monoclonal antibodies. It depends on our application, our use, our purpose that I will be highlighting in my talk. So along with, we have Dr. Chandresh Sharma, who has immense experience in making monoclonal antibodies for the diagnostic purpose. So he will be highlighting his expertise on monoclonal antibodies in the field of diagnostics. And what happens when we start our making monoclonal antibodies, the first question is we have to patent these monoclonal antibodies, because these all monoclonal, most of these monoclonal antibody can be commercialized. There can be different application, even for the research use, for the diagnostic use, for the therapeutic use, there are different usage of that. So how to protect the IPR of these monoclonal antibody? People feel that we make a monoclonal antibody and it's very simple. Just go to the attorney and they will file the IPR. This is not the real scenario, because I am working with IPR experts since last 10 years. There are real challenges in making the patent of, filing a patent is not a problem, but taking it ahead to the licensing, granting and the licensing is a real challenge. So Dr. Chitra is with us. She has, I think, more than 15 years of experience in that. So she will highlight how we can protect our monoclonal antibody IPR in that. So along with that, we have technical expert from the industry. So Dr. Romsha Verma is from BD Biosciences, that will highlight. They have a robust microvalve based single cell system where we can have analysis of those single cells. She will be highlighting that. Along with, we have Dr. Hari Srinivasan, who will talk about multiplex suspension immunoassay for the protein quantification purpose. And we have Dr. Shabbir Vishwas, who is actually working with the GeneScript company, which is the leading company who takes care from starting from monoclonal antibody production to the cell line development. So he will be highlighting his experience, how academia and industry can collaborate, how these antibody can be engineered, and all those aspects he will be highlighting. So in the workshop, we have tried to involve clinicians also, researcher also, IPR person also, and the industry person also. So we are hoping that all of you will get a broad knowledge about all the aspects of the monoclonal antibodies in that. So in the date 2nd, that is on the 9th December, we have identified 12 modules. These are the different 12 modules. First, we will be giving the basic of the hybridoma fusion and the demonstration. And second will be that what is the immunology behind these antibody rearrangements. And the second one is the expression system, high yield expression system of these monoclonal antibodies, the manual method for the purification of the monoclonal antibodies, and the high throughput purification of the monoclonal antibodies. And there are some graduate students also has participated in the workshop. So we have designed 2 or 3 modules so they can know how the basic hybridoma cell culture techniques and how the PCR is used for the amplification of variable heavy and light chain gene of the monoclonal antibody. So we have designed all those things, and we have a session on the IPR also by Dr. Chitra and Hema. And again, we have a company expert who will tell how these antigen antibody interaction, their binding, are more important for development of the antibody for the right target, for the right application. So we have persons from the Sartorius who will be giving that antigen antibody interaction. And we have GeneScript team where they have automated system when you are going for the large scale production of these antibodies. So they have a system where you can directly purify these monoclonal antibody by removing the manual errors. So they will be highlighting on that. And we have persons from the GE Healthcare who will be expertise as how we can make good antigenic bait, because a right antigenic bait is more important, and purity of those antigenic bait also matters a lot. So they will be discussing on about that. So we have tried our best to give all the participants at least 3 modules of the workshop, but I am making sure that after the workshop, if any of the candidates really want to get more interaction with the faculty members and the workshop mentors, we have given all the details, and they can also directly mail me if they want something specific in future also. We'll be ready to help and nurture the young scientists in that. So to make the session more attractive, we have here turbo talk presentations from the young scientists and the poster presentation. So that will happen parallelly in the day 2. So we have 3 award categories here, first, second and third. So I will ask all the presenters to take it seriously and present in well way. So we have different prizes for that. And in the different workshop, the workshop mentor will be monitoring all the participants, and they will be selecting some of the participants based on their interaction with the participant, and there will be a surprise gift from each of the module. So students, be ready for that also. You will get some surprise gift also. Okay. And the third and the last and the most important part is that on the day 2nd, we have to follow the biosafety precautions because we are going to work in the virology laboratory. So in the workshop kit, all the participants, we have provided a lab coat, and then masks are there, gloves is there, shoe covers are there, head cover is there. So I really request all the participants to please maintain the biosafety regulations in the laboratory, and if they need any help, we have a help desk also. So you can contact our help desk members, or you can directly reach me if any help you needed. So from the IAV team, now I welcome to all of you so that you can join our workshop and take part actively in that. So thank you.
Thank you for that briefing regarding the 2 days hands-on training on monoclonal antibodies. And now to wind up the inaugural session, I invite Session Officer Mr. Ranjit R.S. Nair to deliver the vote of thanks.
A very good morning to one and all. On behalf of Team IAV, I am very glad to be standing here to express the vote of thanks. As all we know, IAV started functioning in the year 2020, and IAV is organizing a workshop and hands-on training for the first time. And this workshop is organized in such a way that all the participants can be sensitized on the technology of monoclonal antibody development and hands-on training on monoclonal antibodies. Here I would like to especially thank our beloved director, Dr. E. Srikumar, for truly inspiring, guiding and supporting us to upbring this institution as a global center of excellence in the field of virology. Thank you, sir. Due to time constraints, I am not going in detail. I would like to express our heartfelt gratitude to our esteemed guests, our speakers, invited speakers, chairpersons of each sections. Thank you, sir. Further, I would like to extend thanks to all the participants, firms, industrial partners to participate in the event. Further, I would like to express sincere thanks of IAV to Government of Kerala, Science and Technology Department, ICMR, and all the sponsoring agencies for financially aiding us. Finally, I would like to thank Dr. Rajesh Kumar, Principal Scientist, IAV, who made this day possible. Thank you, sir. Thank you, and thanks to your team. As all the participants are eagerly waiting for the upcoming brainstorming sessions, so I would once again thank each and every person present here. Thank you, sir.
So officially we are at the end of the official inauguration function. So we are going to start our lecture series next. So we'll be having a 5 minutes break to arrange the, we are having our online speaker Yeah, he's already available. So we are requesting to remain seated for 5 minutes so that we can start the next lecture series.
Yeah, you have to enable me to slide sharing, screen sharing.
So we are officially initiating our first session of the lecture series, which will be chaired by Dr. E. Srikumar and Dr. Abdul Jalil. Dr. E. Srikumar is the director of Institute of Advanced Virology. He was working as a scientist G in the Rajiv Gandhi Center for Biotechnology before taking charge as a director. Under his leadership, IAV is progressing to its prodigious future. Dr. Abdul Jalil is working as scientist G in Rajiv Gandhi Center for Biotechnology in the Cardiovascular Diseases and Diabetes Biology Research Program. He is leading the research on MS-based metabolomics and type 2 diabetes risk factors over time in insulin resistance. Over to you, sir.
Good morning. Welcome to the first session of today's lecture series. So we have two speakers in this session. First one is Padma Shri Professor Dr. Lalit Kumar, and the second one is Dr. Devendra Saigal. So,
Hello.
sir,
You have to enable me to screen sharing. It is still disabled.
Dr. Lalit Kumar is the chairperson of Oncology and Bone Marrow Transplant Department in the Artemis Hospital, Gurgaon. He was the former professor and head of the Department of Medical Oncology, AIMS. He graduated in medicine from Sarojini Naidu Medical College and did his higher studies from Adyar Cancer Institute, Chennai. He has led multiple phase 2 and phase 3 clinical trials and is an eminent figure in immunotherapy. The Government of India acknowledged his contributions for developing low-cost medical facilities by awarding Padma Shri in 2014, and in 2008, the Government of India honored him with Dr. B.C. Roy Award, the highest award by the Indian government in the medical category. We are really delighted to have you on for this occasion, sir. We welcome you to IAV.
Thank you so much. Good morning, and I hope you are able to listen me. Can you enable me to screen sharing so that I can see slides?
Sir, can you see us? Are we visible to you?
Yes, now I'm able to see it.
Okay, sir.
Okay. So that's the. Okay. So, are you able to listen me now? Am I
Yes, sir.
audible?
Yes, sir.
Yes, sir. Yes, sir. We
Yes,
can listen.
yeah.
Okay. So, very good morning to you all, and thank you, Dr. Rajesh, and thank you, Professor Sri Kumar, for giving me this opportunity. So, what I want to do for the next few minutes to give a little flavor as a clinical person that how monoclonal antibodies have really changed our life in terms of the patient's treatment and also in terms of the diagnosis, how it is really happening. Before that, I'd just like to how do I change my slides? Okay. So just to give a little background, the communicable diseases that accounted for almost 61% in 1990. And today they are just less than one third, almost 32%. And non-communicable diseases, they account for almost 50%. And these non-communicable diseases, apart from virological conditions, cardiovascular diseases, respiratory, stroke, cancer has become one of the major problem health from India and globally also. Now, if you see this cartoon here, China leads highest number of cases in the world, followed by US, and India is number 3. And we get most more million new cases every year. So India accounts for almost 64% of all new cancer cases in the world. And just compare like the various common cancers among this. In India, over lung cancer is the most common, and followed by oral cavity, stomach, colorectal, and pharynx cancer. If you compare in your state, Kerala state, lung followed by prostate, mouth cancer, liver, and stomach cancer. Similarly, in women, if you see that breast cancer is the most common, followed by cervix in India, LA whole, followed by colorectal, ovary, and oral cavity. While in Kerala, you have the breast cancer as the number 1, followed by thyroid cancer, which is very well known. The thyroid cancer is very common in Kerala, followed by uterine cancer, cervix cancer, and cancer of ovary. So this reflects that there have been changes from one place to another based upon some of the common etiological factors, which may be now. The another issue we come across while managing cancer patients is the advanced stage of disease. Hardly less than 20% or 20 to 25% people come in early stage disease, whether it's a breast, GI cancers, head and neck cancer, or lung cancer or cervix cancer. Most people will have either almost nearly 60% will have locally advanced disease or stage 3, what we can call it, and about nearly 10 to 20% will come with the stage 4 disease, which really makes the problem much more complicated. And ICMR has estimated that for all of us, in India, we have like everybody, like 1 out of 9 persons will develop cancer in their lifetime. So this is like an estimate, and for different cancers, like you can see on the left side for the men and on the right side for the women, like breast, cervix, ovary, uterine and lung cancer. These are the kind of risk. So for, in general, 1 out of 9 people are expected to develop cancer in their lifetime. Therefore, this is really one of the major health challenges we all are going to face, and maybe next 20 years or 30 years time is really going to become much more major health problem. Now, if you see this, almost 1/3 of the cancer today are related to tobacco, as you have seen in the list which I showed for men and women. The smoking or tobacco form and without alcohol, they are responsible for almost 1/3 cancers. They are clearly preventing cancers. And another 1/3 are because of unbalanced diet, which we have last 2 decades or so. We have been kind of using a diet which is really unhealthy, and that is responsible for this. About 20% of these cancers are related to the infections. This cancer is one of the very example, and followed by hepatitis causing the cancer. And then on the another part, about 20% may be related to the hormones, especially the breast, uterine cancer, ovarian cancer. We have and prostate cancer have the hormones as one of the important factors contributing to the causation of these disorders. Now, before I really go to antibodies topic, let's give a tribute to this gentleman, Paul Ehrlich. He was a German physician, he was a German scientist who in 1908 awarded a Nobel for the work which he did, and he was the first person to chemically synthesize the for this. And let me just remind you, syphilis was one of the most important health problem in the whole of Europe, and the first which was synthesized from the urea was silversand and by a gentleman. And that time, while attending a meeting in Europe, was coming back in a train, he wrote in his notes, at some near future, we will have a drug will be like magic bullet. And you can see the German word used, and this is what we really know, that monoclonal antibodies are really kind of magic bullet. So let us see how it really has gone. Subsequently, after 4 decades, 1975, 7 decades later, in 1974, Kohler and his team, they received Nobel Prize for on the hybridoma technology, which was the basis for the development of these monoclonal antibodies. By the year 2020, we have 570 monoclonal antibodies which were under clinical trials, and 79 of them have been approved for clinical use, and of 79, 30 are for the treatment of the cancer in various different kind of cancers. So if you see this chart here, in this cartoon, you can see on the left side the market value of these molecules which really have been developed 75 till year 2020, and the names which are in red color, these are the most selling molecules of monoclonal antibodies, which includes the rituximab, that's the monoclonal antibody for the non-Hodgkin's lymphoma, which I will be talking to you just in a second. Then we have the rituximab for breast, which is one of the important problem, as I just showed you, that one cancer. Then we have checkpoint inhibitors, which are currently being one of the important discovery of the last 5 years. And some of the companies which are making it, the Genentech is one of the company, which almost nearly 30% of the all this monoclonal antibodies market share. And these are the different technology which are being used for making monoclonal antibodies, starting from the mouse edema, which is responsible from the murine derived monoclonal antibodies, then fast display, which take the chimeric and humanized monoclonal antibody, and transgenic mice, which by making the human monoclonal antibody, and also using the single B cell, leading to the monoclonal antibody. And based on the source of monoclonal antibody, you call them the murine, like you see the both constant and the variable regions are derived totally from the murine source. Then you have chimeric, variable regions are from the murine, and you have humanized. The word JUMEP, that's the in the and the JUMEP comes the molecule, and the variable segments are derived from murine source, and you have the completely that is a UMAP. So that is the word which comes in the end in the name of the molecule. So before I really go to the monoclonal antibody, another thing which I always were fascinated by this Philadelphia story. I'm sure most of you are aware, Philadelphia chromosome was the first chromosome aberration detected in the chronic myeloid leukemia in 1958 by Noble Hungerford. Subsequent work in the around 1999 and year 2000, on the bottom side of the picture, you can see the Professor John Goldman, then Drucker, and they developed this molecule so-called imatinib methylate. And this tool had changed this very small molecule. So this is the imatinib methylate, which is in kinase inhibitor. By replacing or occupying one of the site from ADP to ATP, it prevents the phosphorylation, therefore prevents the cell proliferation. And that drug has become the most wonder drug, the first targeted therapy which was introduced in the treatment of cancer in the year 2000. So similarly, along with monoclonal antibodies, some of these small molecules came in the picture, and this became the one of the poster for this treatment of chronic leukemia. And as you see in the on the top, this is some of the data which we placed from our data, that we had the complete response and the overall survival almost 6%. In almost nearly 200 patients, which are the first frontline imatinib methylate. And you can see on the bottom, this is one of the national data from Germany. You can see the survival used to be close to 10% in 1980s or 2000. Subsequently, with the time, the survival to reach 90% at 10 years. The mean survival for chronic leukemia is 5 years. When we were students, we entered in the field, used to be 8 to 9 years, 8-25, which is almost equal to the people same age in the community. For a normal person, survival is most parallel to survival what we see in the today. So that has been one of the most sad story, and the beauty of this molecule, this is the oral medicine which has to be taken once a day and very, very minimal side effects. So this has really changed whole how we really look. And parallelly, we had the monoclonal antibodies which were developed, though even story started in 1975 of hybrid technology, but many of these molecules came almost same time. So, giving the first example of the anti-CD monoclonal antibody or rituximab, chimeric monoclonal antibody, and used for B-cell non-Hodgkin's lymphoma. Next slide. So now this you can see this is the first paper which was published pivotal study in New Medicine in 2005 and 2003, which showed that if you combine the monoclonal antibody R, that is rituximab, along with the chemotherapy combination, which is CHOP, which is standard non-Hodgkin's treatment for Hodgkin's lymphoma for almost last more than 3 and 4 decades. And it still continues to remain so. And it has clearly shown that both event-free and overall survival was superior when you combine the rituximab along with the chemotherapy. So this is the first important advancement which took place in the treatment of non-Hodgkin's lymphoma in the almost in 3 decades. Okay. Fortunately, in the story which was covered in the hematology, you can see the top line, blue line shows the patients who have been treated with rituximab, and the red line shows the patient who did not receive rituximab. And in India too, the generic versions of rituximab, or we call them biosimilars, were derived and which have shown the almost parallel survival both in terms of overall and the progression-free survival. Mabthera was the original and Eritux was one of the generic molecule or biosimilar by one of the company. And the trial in a good number of patients have shown that the survival was similar between the two. That has become one of the success stories that Indian industry, pharma industry would come up with a similar kind of development, good quality drugs and changes which can lead to the improvement level. Another in a hematological condition, another antibodies will be today are using routinely is the myeloma. In multiple myeloma, there's a deficiency of plasma cells which are B cell origin and these plasma cells, they do express 38 on the surface, and daratumab is one of the body against this CD38 and being used in combination with the newer drugs like lenalidomide and dexamethasone, or bortezomib, lenalidomide and dexamethasone, so different combination. Similarly, the another monoclonal antibody bortezomib, which target the SLAM epsilon, and rituximab is another monoclonal antibody by another recently one of the company anti-CD38 monoclonal antibody. These are currently being used mainly patients with refractory myeloma, and all last 3 years daratumab has been approved for the first line treatment of multiple myeloma using the 3 of the agents monoclonal antibody. The other, let's now bring it to the breast cancer. Now in breast cancer, I know that we have two gene, which is one gene expressed on the surface of breast cancer cell in almost 50% of breast cancer patients. And they are the molecular type cancer. You can see the dark arrow on the patient who have the HER2 gene, and if they do not keep with the HER2 mutation by the trust, then this becomes very poor. And today, now have the drugs available for this, And as a, so the first which in the year 2005 and became the one of the pivotal study called TIL, which showed that in 1 year of the monoclonal antibody completing the treatment surgery, therapy and radiation for breast cancer patients who were expressing new gene, then your survival, disease survival is significantly higher. And based on trial, this drug monoclonal antibody has been approved and now today considered as one of the standard of for the breast cancer women express who express the HER2 gene. So it is mandatory today that for all breast cancer patients, whenever we take biopsy, we not only have to demonstrate pathological area, invasive ductal carcinoma or lobular carcinoma, then we also have to have the information on whether they are estrogen progesterone receptor positive or negative, and also HER2 positive or HER2 negative. And so this was distant disease-free level. Again, this was also superior by the patient give the trastuzumab again for the patient expressing. Now, based on the trial, mandatory develop some areas based on histochemistry and also on the phage. What this study found out that patients who had the 3+ clearly, they immunohistochemistry, they were called renewed positive. Those were only plus or based number of cells and staining, they were called HER2 negative, and those who were 2+ were so-called equivocal. And became the guidelines were issued that all patients who were HER2 positive 2+ should undergo the FISH test. Right. So these are the guidelines, and according to patients who are positive on chemistry should undergo FISH test, and based on the ratio of the HER2 signal divided by the signal at chromosome number 17, and based on this ratio, them they are HER2 positive or HER2 negative. Those who are HER2 positive obviously should receive the full treatment for this trastuzumab. And enough molecules also have been developed today for the patients who are HER2 positive breast cancer. Next slide. Now, another similar story is for the vascular endothelial growth factor. Now, many of you may already be knowing that VEGF is necessary for the normal physiological development of the vascular channels in our body during the development process. So, it has been found out that the patients like ovarian cancer, colon cancer do experience VEGF, and antibody against this VEGF1/4, there are 4 isomers are available. So anti-VEGF antibody has been shown that this can be prove the outcome for these patients, those who have the VEGF positive, or we normally for the patients of ovarian cancer, we give the with the therapy, we also add the anti-VEGF and called bevacizumab. So this is again humanized monoclonal antibody which is combined along with the chemotherapy. Next slide. So these are the 3 pilot trials again on the top two, they are the gynecology of US and I group from EU, and they have clearly shown if you combine bevacizumab with BEGF in the treatment of ovarian cancer first, along with the chemotherapy and regimen, progressive survival benefit. And also on the bottom, this is the study in the patient with the recurrent ovarian cancer line can combine this molecule along with the chemotherapy, there is benefit in the progression-free survival. So this again has become now one of the standard of the management patient. Now coming back to the most exciting work been done in the last years or so, and for this work, the Alicia and Propanjo, they received the Nobel Prize in 2008. And this basically work is that immune checkpoint inhibitors, normally the T cells are responsible for looking after our any foreign antigen or any cancer cell in our body. Somehow, because of the tumor, they're able to express molecules and able to block or develop a sort of natural break on the, and therefore the T cells fail to recognize or fail to act on these tumor cells. So what they developed this concept that making cells to recognize these tumor cells and attack them can improve the outcome. And some of the molecules, the PD-1, which is programmed cell death 1 and programmed cell death ligand, against these CTLA, but in the last, this again has very field which is very exciting and huge amount of research is being done to develop molecules, and I would say that you can find out a new monoclonal antibodies. This immune checkpoint inhibitor has been claimed that this effect. Some of the new molecules which are being shown here in the table, ipilimumab, which is one of the drug of choice for treatment of malignant melanoma, which is the totally outlook has become different now. Similarly, nivolumab, commonly used for the treatment of lung cancer, urothelial system like ureter and kidney cancer, and then the drugs atezolizumab, all being tried as cancer and in the again lung cancer. So these are exciting. So this is one of the study, the lung cancer recent stage which were able, after the randomization, they were given chemotherapy and with this monoclonal antibody. And what shown very that the progression-free survival was significantly superior. At 5 years, there were difference of almost nearly close to 17%. Now, this is some unprecedented lung cancer, especially advanced disease. There few people will be living beyond 5 years. You can see there are number of people who are surviving with the help of this monoclonal antibody. And this is the overall survival. Almost nearly at 5 years, nearly 43% were alive in the group which received the antibody, immune inhibitor. See, another study in the bladder cancer, again patients who were susceptible or locally for metastatic urothelial cancer, and after the initial chemotherapy, randomized to either receive the antibody or only best supportive care. And what again they have shown that the median overall survival was superior if those patients who received compared to those who received only chemotherapy but no antibody. Similarly, the progression-free again was almost 15%. The risk of relapse was lower in the patients, the group which received the monoclonal antibody. Next slide. And
Sir,
rectal cancer, this is something which was very much in all newspaper media. They have found a wonderful drug for the treatment of rectal cancer. In the month of June and July, all papers were full of this, and we used to be getting calls from our patients that what is this new drug? I also get particular so that my cancer can be cured. Now this is the molecule dostarlimab monoclonal antibody, which was used in this study, a small pilot study for rectal cancer for patients who were stage 2 and stage 3 and the mismatch repair gene deficient. This makes these patients a little more prone for at high risk, so-called high risk. So they receive monoclonal antibody along with the chemotherapy and radiotherapy.
Sir, could you wind up in 2 minutes? Please?
Okay, just 2 3 more. So you can see this on the, it's like 14 patients were able to clearly show that all patients achieve complete response. This is on the last column, you can see that. So really something very, very surprising to everybody. The drug was so effective, they could achieve complete response, and these patients do not require any surgery. Those who underwent, they were pathological complete response. No evidence of cancer in the pathological specimen. Next slide. So, next slide. So then again, like who are those patients who really benefit with this particular molecule? And now this we have been able to work out that the tumor burden calculate, then the patients who have the high tumor mutation seem to benefit with more immune checkpoint compared to those with the tumor burden. So that has become the important thing. And recently, last month, people from the Tata hospital, what they have used the low dosage of immunotherapy. So far, all this data has from the Europe, the US, and the drug was used much higher, which is very expensive and very few can afford in the developing countries. So they have used almost like 1/3 of dose. They could, along with the compared with the chemotherapy, and what they have shown that the low dose also. I think the slide. The overall survival and progression-free survival compared to only chemotherapy versus those who got chemotherapy plus low dose, they have been able to show that in the low dose also works, and this is a good news says that it becomes affordable to many more patients compared to those if you use the initial which was the higher. So currently, let me just summarize. Using immune checkpoint to inhibit the early stage of cancer is one of the idea which is being now currently worked out, and whether to more than molecule with more than one target, that is something people can work out, and with the mechanism of action, because obviously people are made developments, and how to prevent the toxicity or minimize toxicity, which also is one of the issue. Means many patients can have this toxicity, and can we find some of the biomarkers of response? So that is something. These are some of the things which currently people are working. I think I will just stop it here. I had some few slides on CAR T cells, which is another exciting area in the field of hematology. But maybe in lack of time, I will just close it here. If you have any questions, I would be happy to answer. Thank you so much.
Thank you, Professor Lalit Kumar, for having a wonderful overview on the use of monoclonal antibodies in cancer treatment. One of the interesting things that we could notice was even the biosimilars are performing equally or better in the treatment. So that is a very encouraging news. Rather than depending on the primary product itself, we can have biosimilars of equal or more efficiency. So we can take one or two questions, and maybe then we will proceed to the next session. Any questions, please?
Thank you, sir, for the wonderful talk. So actually, in your talk, we found that the immunotherapy has really played an important role in the cancer therapy. So what is the stage you think, stage 1, stage 2, stage 3 for cancer, where these antibody can play a major role? Because means importance of the monoclonal antibody and how the diagnostics play an important role. At what stage we have to give the immunotherapy and all those things little bit.
Okay, so this is a very important question. In fact, now when we see in the patients, they come, they think that it's something new drug, and this is a bundle drug. So can we get immunotherapy? So the answer is that not all needed. Only patients who have failed as of now, who are getting in line of third therapy, immunotherapy used. One. Secondly, recently there are 2 papers have shown for patients of cancer, for along with the therapy, if immunotherapy is stage 3 disease, then this is something which improves the outcome. And all for the esophageal or food pipe, again, have shown that after initial surgery, if you give them as a adjuvant chemotherapy and immunotherapy, then the result is better. There are some of the new papers which have come in the last 1 year. That's what I said. These are something which we really need to after showing the efficacy in the disease, the field is moving to bring the efforts are to bring into the early stage of disease without compromising the on the part.
Okay. And the last question is that in your talk you mentioned that in Kerala we have more thyroid cancer cases as compared to the other states. So do you think it is related to some environmental factors, genetic factors, or maybe some studies is there on that can highlight why these thyroid
I think thyroid cancer people lot has been done, and what they have shown that probably the uranium content was higher found in the area. I think that probably has been attributed one of the factor responsible for a higher incidence of thyroid cancer in Kerala. Similarly, data has been reported from the northeast part, especially Meghalaya and Arunachal, where they also have the higher content of some of these uranium product.
Okay, yeah.
Thank you.
Anyone has any question? Okay, can I have a quick question? Expecting a quick answer, sir. In the initial introductory slide, you said that 1/3 of the death, cancer death, is caused by high glycemic carbohydrates and physical inactivity, etc. My question is it only related to cancer death, or does it have any relationship with the cancer onset? High glycemic carbohydrates.
So I think the question was like 1/3 of cancers are called because of lifestyle related.
No, it was, yeah. You mentioned it was related to cancer death. So is it the same reason for the cancer onset also? That was the question.
No, I'm not sure if I was correct. It was something.
Okay, sir. We will interact some other time.
Communication issue was a little there.
Thank you, Padma Shri Dr. Lalit Kumar, for this wonderful session. And we are moving on to the next speaker. That is Dr. Devinder Segal.
Okay. Thank you so much.
Yeah.
Thank you so much.
I request the students to participate, interact maximum. Please have questions actually, especially students from RGCB. And we will have more questions if you can finish your talk within 20 minutes. Yeah.
So the next talk of the session is given by Dr. Devinder Segal on the topic Immunology Behind Antibody Rearrangement. Dr. Devinder Segal is a senior scientist at the National Institute of Immunology, New Delhi, where he heads the Molecular Immunology Laboratory. Dr. Segal did BSc from Delhi University and MSc and PhD from the School of Life Sciences, Jawaharlal Nehru University, New Delhi. He moved to National Institute of Allergy and Infectious Diseases, National Institute of Health, USA for his postdoctoral studies. His recent studies are focused on identifying the molecular features of antibodies that are responsible for conferring protection against the human pathogen Streptococcus pneumonia in vivo. We welcome you, sir.
Let me thank the organizers for inviting me, and especially Professor Sri Kumar and Rajesh. I'm absolutely delighted to be here in Trivandrum. And so after this session regarding use of monoclonal antibodies in clinical session, I would try to help you understand a little bit about how this would apply to infection setting. And what I will be describing to you is how what we can learn from these monoclonal antibodies, and I will share some data along these lines from my laboratory that will help you understand some of the aspects of the monoclonal antibodies. Okay, so I work in the area of molecular immunology, and we use a bacterial pathogen called Streptococcus pneumoniae as a tool to perturb the immune system and try to understand how the immune system sees this. So this Streptococcus is a gram-positive encapsulated extracellular bacteria, and the form of protective immunity is primarily antibody mediated. That's what connects it to the story that we will be discussing here. So we have been looking at various molecules that are present on the surface of this bacteria, and with the hope that we will be able to understand how it interacts with the host immune system as they may be involved in virulence and pathogenesis of this bacteria. So we, for today's presentation, will be focusing on a particular molecule called PSPA, and I will just describe that a little bit as we go along. So we have work in our laboratory. We are looking at multiple aspects, and one of them being how the host immune system sees a bacterial pathogen. And we also have been looking at various, pathogen immune evasion strategies that are used. We have some interest in looking at vaccine potential of various proteins that are present on the cell surface. We have been identifying new virulence factors, and also how these virulence factors induce alteration in the host and cellular immune responses. So this molecule, pneumococcal surface protein A, was chosen for the reason that it is surface accessible, and it's expressed by all strains of Strep pneumoniae. And what interested us was that antibodies against, if you make antibodies against this particular protein and you passively transfer this into a mouse model that we use for our studies, and if you challenge these with virulent pneumococci, they would be protected, meaning they will survive the challenge. Alternatively, one could use them as if you were to use recombinant protein into these as immunogen, one could also protect. Okay, and more recent data from another group has indicated that when human volunteers were given this recombinant PSPA, the antibodies that were generated in human volunteers when transferred to the mouse system, and if the mouse was challenged, they were protected. So these were the basis of our reason why we were trying to understand what is there. So what I will be doing today is trying to tell you about the antibody response that is generated against this particular protein. Okay, one of the questions that we were trying to answer had to do with the fact that whether the immune system sees the pathogen, the protein, the same way, a soluble protein versus a protein which is given in the form of a particulate antigen. So basically, we simply cloned and expressed the extracellular domain of the PSPA molecule, and we purified it. And we, in order to have a closer look at how the immune system works, we made monoclonal antibodies against in two formats. One was we gave recombinant protein, and in another case we used the whole heat-killed bacteria, pneumococci, for making these monoclonal antibodies. So we, had got 36 different monoclonal antibodies that were there. And as one would expect from a response against the protein, we have it's a very polyclonal response, and what you would find is that when we looked and sequenced the transcripts for the heavy and light chain antibody genes, we found that they were coming from a variety of different families that were. And for people who have some understanding of how the structure of the antibody molecule looks like, there are parts of the protein which make antibody molecule which make contact with the antigen, and those are called complementarity determining regions, CDRs. And a certain one called CDR3 is the one which is responsible for, is the major contributor with regard to how they towards the antigen binding and specificity. And what we landed up seeing is that in the panel here, I can see that. Okay, so we have molecules which had a certain length of CDRs. There is the pointer. Pointer. So what we found was that that a CDR3 length of a certain size were expressed in higher numbers. And again, I would not go into the details of it except to tell you that typically during VDJ recombination, we have a V to D rearrangement as well as a D to J rearrangement. In this particular instance, what happened was that we have about 20% of the monoclonal antibodies had a heavy chain where we did not find any DH gene encoded amino acid, which was very surprising. And so this was an interesting observation I thought I will share with friends in the room. And so essentially, once we had these monoclonal antibodies, we wanted to find out what within the molecule, where does it bind? Okay, so essentially trying to see if we can narrow down to the regions which are responsible for where the epitopes are located. And in here, what you see here is the not all parts of the any given protein would elicit antibody responses, but so we were trying to find out where does it bind. So the approach that we took was to make a series of overlapping and non-overlapping fragments shown in yellow here, and we have purified and expressed this protein, and then we try to map, localize the epitopes that are there using these monoclonal antibodies. So this is data from a particular antibody as a representative. So essentially we used Western blotting and dot blot analysis. We have used recombinant protein and lysates from pneumococci as to see where they bind. And on the right hand side we have a series of dot blots which corresponds to the various fragments that we have used, and based on this analysis we are able to figure out that which region within PSPA is this map. And in combination with the assay which is called ELISA additivity assay, we were able to find out whether these monoclonal antibodies see the same epitope, or are they seeing epitopes which are distinct. So again, in here on the left-hand side, we have grouped these antibodies. So in this assay, each antibody is made to compete with all the other 35 monoclonal antibodies. So there is a lot of experimentation that has gone into this data. So within the same group, for example, let's say we are looking at group 7, the antibodies actually compete with each other, suggesting that they are either topologically identifying the same epitope, or they are having overlapping epitopes, but across groups they do not compete for binding. So we have a whole bunch of, so essentially overall what it is saying is that there are at least 12 distinct epitopes that may be present in this molecule. Okay, so one of the other considerations that I was mentioning is that here we are doing a comparison of the relative avidities of the antibodies that are induced in response to immunization of mice with recombinant PSPA, and it's being compared with the avidities that are generated in response to the particulate antigen, that is the whole bacteria. And we found that the avidities are, you induce much better avidities when you are using recombinant, when you're using the antigen as a particulate antigen. So keeping these things in mind, what we have, we narrowed down 36 monoclonal antibodies for the kind of experiments that I will be just describing in a bit. Are too many to deal with. So we shortlisted them based on the antibodies that were generated against the heat kill bacteria, because they were making better antibodies, and also the fact that they were of IgG isotype. Okay. So the question really is that if you have an antibody, are we in a position to predict whether these antibodies are going to confer protection in vitro? So if we can do that, it saves us a lot of effort. And so we made some beginning in terms of trying to figure out, trying to elucidate the molecular attributes of the protection conferring antibodies. Okay. So obviously, as I mentioned, we have localized the epitopes for these antibodies, and we were interested in finding out which of these antibodies confer protection. Okay. So what you see in green are the ones that we'll be interested in, because they are recognizing the epitope which induces antibodies against. Just to add a little bit, this particular protein shows serological variability, which means that between strains there are slight differences in the sequence, and which can be recognized by the immune system as being different. And so there are 3 different families to which this PSPA molecule has been classified, which translates into 6 different clades. And so each of these PSP of each of these clades have been purified and in the lab. And we have looked at their ability of each of these antibodies, 18 of them, to bind to pneumococci of different clades. Okay. So what we have seen is that these antibodies bind to members of family 1 and not the others. And the primarily that was the reason for it is that the PSP that was used as the immunogen itself was coming from this family. So we then subsequently went on to look at how, because the important thing here is that pneumococci is an extracellular bacterial pathogen, and as I indicated earlier, the protection is antibody mediated. And so if we wanted to make sure that the antibody that we are looking at, the monoclonal, actually binds to the surface, if the epitope is accessible to the antibody. So these are flow cytometry analysis of pneumococci, and we are looking at the ability of the monoclonal antibody to bind to the different strains of the clades that we just mentioned. These are examples from 7 different strains. And as you can see from the open bar on the right, it is indicating that there is, it binds very well. And then we looked at more number of strains just to make sure that these antibodies are broadly protective. And again, this is data using 6 different, from 3 different strains from each of the family members that I was mentioning, and we then chose 8 antibodies for more detailed analysis. Okay, again, one of the ways that the immune system clears pneumococci from the host is what is called opsonophagocytosis, which involves binding of the antibody or the complement on the surface of pneumococci. And there are receptors present on phagocytic cells, which are basically neutrophils and macrophages, which are then that they will bind and endocytose, take up the bacteria and then kill it. Okay, so essentially we were trying to follow the ability of the antibodies to fix what are called complement present in the blood on the bacterial surface. There are other pathways that are also independent of the antibody, but this was something that we were interested in, and we followed it up. So from again, this is flow cytometry data, which is basically telling you that I've just taken example of one antibody. These were done for all the antibodies that I was mentioning, and we can see that there is a lot of, beautifully, the antibody is able to induce the binding seen by the right shift of the curve. And so again, this is again summary of experiments from a lot of different antibodies and different strains. And what we found was that these antibodies are indeed able to fix complement, and the extents are a little different. So again, pneumococci is an airborne infection, so typically it is taken up by aerosol, much like COVID that you people have all seen and experienced. And then it goes into the nasopharynx. From there it can then migrate into lungs to cause pneumonia, and then it can then subsequently go into the bloodstream causing sepsis. So in order to mimic that experiment, what we have done is that we've taken the bacteria and incubated with these antibodies that I was just referring to, monoclonal antibodies, and then put the pneumococci in the coated antibody in human blood for different periods of time, and then go back and try and see if, how many bacteria were killed. This is basically followed by a simple experiment which involves just plating the bacteria and seeing counting the number of colonies. And again, just to, in the interest of time, just like to summarize this, saying that, these different 8 monoclonal antibodies were able to, induce killing within the bacteria. So they had bactericidal activity. The extents were a little different, but again, all the antibodies were done this. We have done this using multiple strains, and they show, good bactericidal activity. And with this set of in vitro data in mind, then we moved on to doing experiments with what is called mouse passive protection experiments. These are experiments done in a biosafety level lab 3 primarily. And here what we are seeing following is on the x-axis is the survival of the mice. So the experiment is that you take mice, you purify the monoclonal antibody, give it intravenously, and then you challenge the bacteria, challenge the mice with the virulent bacteria, and you observe these animals and see how whether they survive the challenge or how long do they survive. So based on these assays, we have, again, these are challenge experiments. If for instance, we are looking at panel B, if the antibody is working well, then animals will survive till the end of the experiment, which is day 21. And as you would see, let's say on the panel on the second column on the right, B, the parallel lines on the top, you see this is telling you that all the animals survived despite a challenge with the lethal dose. Okay, so these experiments were done by giving 5 micrograms of the purified monoclonal antibody, and then we did the experiment again with much lower dose to see what would work and what would not. And again, what we notice is that these antibodies are doing amazingly well with regard to, to conferring protection. So these are, very antibodies which confer protection, okay, to the animals. It's much like what the antibodies that were being described previously in Dr. Lalit's presentation, but that was in the human system. These are experiments of a similar kind. And so essentially what was the carrier message that is emerging from here is we were trying to figure out what properties should an antibody have or does have which will confer protection. Okay, so for here is a summary table which basically talks. We have done surface staining, complement deposition, blood bactericidal assays, and then mouse survival assays up here. And we are again trying to see what matches. Okay, what was the property or properties that were required for the antibodies that confer protection. And what we see is that we have four sets of antibodies on the right. These were doing a little better than the others, and so it turns out that the protection correlates with the ability of the antibody to deposit complement on the surface. That's the property which correlates with protection. Although we did a variety of experiments, we also went on to look at the relative affinities that by surface plasmon resonance, and the ones which were giving protection and the ones which did not give are not terribly different in terms of their affinities. All these antibodies are doing, have very good affinities. Okay, so it's the ability to induce complement which is the important part. So trying to close the presentation today. So I showed you is that we raised 36 different monoclonal antibodies. We mapped the regions to which these epitopes are that bind to this antibodies were present, and then we did a variety of in vitro as well as in vivo experiments that were helping us figure out which of these antibodies confer protection. Those are the ones that are present in green. And so now the question that we were trying to look at is that what is, remember when you put in a vaccine or antigen, you will be making a lot of antibodies. Only a fraction of them are useful as far as protection is concerned. Rest of them is kind of waste, and sometimes it could be dangerous for people. Okay, and typically seen for many the presence of antibodies which are non-neutralizing can actually worsen the subsequent episodes of the challenge of vaccines. So we are trying to figure out what are the epitopes that are being recognized by these protective antibodies. So those are the ones which would be interested. And why are we interested? The idea, as I was just indicating, that only those antibodies, the epitopes which can induce protection-inducing antibodies are the ones which are good. So if you map them and you can kind of stitch them together in two different formats that I've just indicated, in green lower down here. So those are the ones which would then be inducing, the hope is that you will be inducing only antibodies which confer protection and nothing else, okay? And in that way, your immunogen could be, very, very potent. And so in terms of future directions, we're trying to map the epitopes of these protection conferring antibodies, and also to try to, once we have we have identified the protective epitopes, and then maybe we can just simply synthesize it and use that as an immunogen, and hopefully they would give those protective antibodies. And to close, these are the team members who have contributed to the portion that I just described: Soma, Devjani, Sohail, Parul, Naeem, and Rais. And the work has been funded by the Department of Biotechnology and the NII Intramural Program. So I will close here. I will be happy to take any questions, and thank you very much for your attention.
Thank you very much, Dr. Segal. Actually, this is a very ideal and apt presentation for a workshop like this monoclonal antibody, because you just traditional like hybridoma technology you have showed, starting from the fusion, cloning, and what is that, functional characterization of the antibody, as well as the even preclinical testing, everything which is shown. I hope that the students understood each step which has to be, one has to follow for a workshop like this. And the session is open for a couple of questions. Please ask.
So thank you, sir, for your presentation. So actually you mentioned that 4 of your monoclonal antibodies are very protective. So epitope of these monoclonal antibodies were linear epitope or some conformational epitope?
Yeah, a very interesting question. See, one of the things that I failed to mention was that the extracellular domain of PSPI is a coil-to-coil structure. And this is an experiment which is a simple experiment to do, wherein you take the recombinant protein which you already have in the lab, and you simply boil the protein and see if the reactivity is lost or not. And as we did this experiment, we boiled it for 10 minutes as we would do for SDS page experiments, and then and we went back to see the reactivity, absolutely no difference. Okay, zero difference. We also then did this experiment we boiled it for like 2 hours, no difference. The conclusion one would get is that it's maybe linear epitopes. We did the experiment with polyclonal sera as well, no difference. So structural biologists landed up telling me that the coil to coil structure is an extremely stable structure. You can boil the protein for any amount of time and they will snap back exactly. So we have not been, I can't tell for sure whether this is linear or conformational. We had initiated some experiments where we are doing, making overlapping small peptides at 15 mer overlapping peptides and then trying to map it. So we are at that stage at the moment.
So I think, sir, usually people think that monoclonal antibody used for diagnostic and therapeutic, but you are so represent the reverse way. By using the monoclonal antibody, we can design a vaccine that can be suitable for use as a therapeutic person. So I think it's very wonderful, and it's a novel kind of thing that can be used. Yeah. It's like me identify the epitope and then make the subunit vaccine.
Then you make subunit, and only restricted to protective epitopes so that you don't, make antibodies which can be deleterious or do not contribute to protection. So that's the idea.
Thank you.
Yeah, Dr. Saigal, it was an excellent presentation. If you simplify these complicated steps, I mean, like in a very simple terms, students will get confused. They will think that it is a very simple technique. You have made the presentation in a very straightforward manner. That was wonderful.
Yeah, I'll have more details for people who will be attending the hands-on experiments.
Yeah, such an elaborate presentation. And so one question was like, how common is this antibiotic resistance in this pneumococcal, and where are you using those strains which are resistant to particular antibiotic?
The last issue of Lancet has India on the it's on the cover, basically saying that this is among the data from India says that this is, the maximum number of children who die from pneumonia in the country is in India, and along with Klebsiella pneumoniae and Streptococcus pneumoniae covers almost everything. So this is a very serious killer, and the topic of that is that of this study which came like last week, essentially was saying that the antibiotic resistance is massive. There are examples of strains which are resistant to 45 different drugs, 45, one strain. So it's a huge issue, and that is one of the reasons why we have been focusing on the vaccine development of it, primarily that drug resistance is not going to be easy to deal with, so you have to have another intervention which may be not dependent on the MDR multi-drug resistance issue. So that's the reason why I thought it's worth our effort to try and look at understanding the vaccine response in a great detail, as well as having leads that can help you develop better immunogens, as well as
Any of these strains that you used was of antibiotic resistance?
So as a matter of biosafety for students who are working, we have made it a point to use something which is sensitive to all known drugs, but there are lots of them that are there.
Is there any mechanism to predict that least like your antibodies will work against these organs?
I don't see any reason why they should not. Okay, because the resistance mechanism is radically different from how it will. So they should work beautifully, and that's the overall thought process that has gone into it. That it is independent of the status of the drug resistance. So that's the idea.
One last question, like only this opsonization is the only mechanism by which it would be acting, or do you think any other mechanism?
See, as I said, if one were to just take antibody against pneumococci, the ones which confer protection, and just put it in mice, you don't need anything else. All you have transferred is just one monoclonal antibody can do it. So that tells you that the role of antibodies is paramount. Okay. The other aspects, for example, TH17 being contributing, people have used, other groups have tried to use a different approach of, making vaccines. But as I said, the evidence is very strong that antibody is the thing. So that's why we thought it's better to follow that. In biology, there is nothing like, only one works. So by and large, that is true. But the reason why we picked up this pathogen in the first place was that it is extremely skewed in that manner. Yeah.
Yeah. Any questions from the audience? Yeah.
Hello sir, my name is Krishnasri. I'm from Government Arts College. I'm a PG student. I don't know, my question is relevant to the topic, where I just had one doubt that what's the purpose of having that molecular structure on the surface of this bacteria? I've just having doubt that if the bacteria goes mutant for that particular molecule, will this will we be able to tackle this?
Yeah. So interesting question. The question is whether if you have a bacteria which lacks this protein PSPA, would this still work? See, we did a clever thing in terms of trying to in the choice of the protein. Okay, the first thing was that there are no strains which lack this bacteria. Okay, so it's playing a very central role in terms of the biology of the pathogen. Not to say that you cannot get rid of it. We made a mutant which lacks this protein, but under clinical settings you don't get anything. So about 50 to 100 strains have been sequenced. All of them have it. All clinical isolates have it. So that's the reason why we picked up. If one were to have a situation where the bacteria can drop the gene and still work, then this would not work. Yeah. Thank you for the question. Yeah. Yeah, please.
Hello, sir. It was nice to hear you. I'm Gaurav, master student from RGCB. In one of the slides, you told that in the presence of alpha PSPA monoclonal antibodies, there is a deposition of complement C3. Can you brief it a little bit?
Can I elaborate on that?
Yes, sir.
See, what happens is there are different pathways by which this, complement components are deposited on, pathogen, surface of pathogen. In this instance, we are looking at bacteria. And one of the pathways is triggered by the antibodies. Okay, so when you, in the presence of the antibodies, the complement, which is soluble proteins present, they then can be deposited on the surface by a covalent bond. Okay, the C3, there is, if you go back and look at the complement cascade, there are a series of steps that are involved, which involve cleavage and then subsequently covalent linkage and stuff like that. So C3b is one of the components which triggers this process, and subsequently you have a C3-9 attack complex which is produced in the membrane. And with the result, there's a pore that is formed in the membrane, and the contents of the bacteria come out. The bacteria then dies. Okay, so that's the assay which we were followed to try and see what happens. Okay, does that answer your question?
Does that it occurs without this antibody?
No, that can also happen, but we have controls to say that it's not zero. As I said, there are other pathways that are there which. But when you in our experiments we use what are called isotype controls, and that will tell you whether what is the contribution coming from it's never zero, okay, because there are other pathways that are involved, but the role of antibody and its contribution is the kind of readout we get from this kind of assays. Okay, but by no means that's the only way. Okay, there are other pathways that are there. Okay.
Thank you, sir.
Any more questions, or maybe we can have more discussions during the tea time? One
Absolutely.
Technical question I had,
sure.
Like you had used the hirudin as a hirudenized blood, not heparin. So any particular reason for?
So we were using human blood for these experiments, and the pathogen has actually evolved with humans over a period of time. And for some of the other anticoagulants that are used commonly, this is a lot more expensive experiment. It interferes with the complement assay. Okay, so therefore we opted for the hirudenized version. That's about it. Yeah.
So thank you so much for a wonderful presentation.
Yeah, pleasure. Thank you.
Yeah.
We are kindly requesting Dr. E. Srikumar, our director, to hand over the token of appreciation to Dr. Devendra Segal. Sir, please. Now I am requesting Dr. Rajesh Kumar to hand over the memento to our chairman of the session, Dr. Abdul Jalil sir. Thank you for accepting our invitation. So this marks the end of our first session for the lecture series. So we'll have a quick tea and come back, but before you go for the tea, we have arranged you a photograph session, a group photo for us all. You can assemble right outside the building for that group photograph, and after that we'll go for a quick tea. That quick tea for the candidates are arranged at the entrance of the registration desk building where you have arrived first in the morning. I hope you know we'll have our volunteers there to direct you to this place. So thank you.
Hey, I am going for a vacation. Stop, stop, stop. I know you want
antibodies, different method of isolation of the antibodies. So, but when we call the antibodies, in between the antigen and antibody, there are two specific entities that are involved. One is the epitope, another one is the paratope. So what is the epitope? Epitope is the region which is present in our antigen, and paratope is the region which is present in our antibody, and these two, antigen and paratope, basically interact to each other. So in the below I am showing some of the differences and some of the basic property, what is antigen and what the antibodies are. So with the antibody and antigen, we also like to understand couple of the terms which are polyclonal and monoclonal. We will gather here to understand what are the monoclonal antibody and how we will be isolating it. So when we talk about the polyclonal antibody, in this figure what I am trying to show that with the conical, circular and square shaped structure, these are the different kind of epitope. And these are basically the ideal condition what a protein or what a antigen normally reflect in our body. This is a polyclonal response, and polyclonal response from a protein which contains all the epitope of the same kind is not an ideal condition. So we have a polyclonal antibody where we get a response from a different epitope, or the monoclonal antibody where all the antibodies are same. So what we do, when we look into a large protein, it contains very different epitopes, and that leads to generation of polyclonal response. And we will be understanding in the whole these 2 days what are the monoclonal antibodies and how we could isolate these monoclonal antibodies. So from the monoclonal antibody, if we come to the small peptide or immunogen, where we have fewer numbers of epitope, but the response still be polyclonal. The ideal condition is that when you have only one epitope, then your response will be monoclonal. But at the same time, we have the option to isolate the antibodies. This is through either by the fusion of the B cell, hybridoma, or by different screening. So here I am just trying to take you all the 3-4 different pathways which you will listen today, how these methodologies are being used in order to isolate the antibodies. But the important thing that need to see over here is that in all the 4 of these methodologies, the thing that is mentioned is the target or the antigen. So when we are moving towards the antibody isolation, our input, my first slide shows you that the input is very important. It's totally depend upon what kind of target and what kind of antigen you are using for the antibody isolation. So our B cells are very smart. It's totally depend upon what kind of antigen you will show to the B cell, it will generate antibody, depends on that. So it's totally into our hand how smartly we design our antigen and how efficiently we can make our B cell to recognize these antigen. So antigen production is a process that nowadays every lab is using it, and there are 2 different methodology by which we produce antigen, either through the prokaryotic expression system or through the eukaryotic. The prokaryotic expression system is widely used in most of the lab around all over the world. And in the eukaryotic expression system, we have yeast, insect cells, and the mammalian expression system. In a hands-on training tomorrow, we will discuss more about the mammalian expression system. But in the prokaryotic expression system, there are some pros and cons. The first thing is that it's very convenient, easy to use, and it could be produced and purified through a very less expensive equipment and reagent, and very easy to scale up. But at the same time, disadvantages are it lacks post-translation modifications. The conformation does not sometimes go exactly what we wanted, so it lacks also the 3D native conformation. And sometimes the protein which are produced through this system lacks the immunogenicity property. But there are still applications. We can use it for the purification of the bacterial antigen, what we have listened in our previous talk. We can use for the purification of non-structural eukaryotic or viral protein. peptide-based antigen and immunogen, and eukaryotic protein with no post-translation modification, as you can see in the case of dengue domain 3 also. So basic methodology for the bacterial expression and purification is all same. We all know it's a simple method that we all use. But I am giving you one of the example of the bacterial pathogen for which the antibody has been proven very successful. In one of the pathogen, which is Pseudomonas aeruginosa, is one of the most widely caused for the hospital-borne pneumonia in the case of patients who are on the breathing machine and those are on the catheters, and especially the surgery or the burn patient. The problem with this pathogen is that it develops multiple drug resistance. So to deal with that, antibodies are something that is the most widely needed, and we are very happy to know that there are large number of the antibodies which are at the different steps of clinical trial. So this is some steps, where actually we need the antibodies for the treatment. We come to the dengue. We know that there is still in India no vaccines are available, and this is something we all suffer every year. In the dengue, the surface protein, which is actually the target for the antibody development and the vaccine target development, is the E protein. E exists on the surface as a dimer. This particular protein has 3 domains, domain 1, 2, and 3. Domain 1 and 2 basically fold in conjugation to each other, but domain 3 is independently folded entities. And we have very lucky to know that this particular domain has been purified through the bacterial expression system, and using this protein, large number of antibodies has been isolated, and these are broadly neutralizing antibodies, which is something that what we need for the dengue in terms of antibodies or also in terms of the vaccine. In addition to that, when we talk about dengue, the NS1 based diagnostic, which normally used for the detection, is also being produced from the bacterial expression system. Again, the next expression system is a yeast expression system. We are moving towards from the primitive towards the advanced stage. It is a slightly advanced expression system than the bacterial expression. Here, the advantages are it produce high yield, stable strain, rapid growth, and that also contains some post-translation modification that were not present in the bacterial expression system. But the disadvantages are also here because the N and O linked glycosylations are different from higher eukaryotes. Also, their glycosylation are sometimes very complex. It goes to the hypermonosylation, and this expression system has its own proteolytic degradation problem. The next one is a baculovirus. Again, a slightly advanced system than yeast expression system. It goes a homologous recombination system where we have a cloning vector and a transfer vector. We all must, or some of you must still be reading about the helper phage, where the vector gets transferred into the phage, it develops, so we develop a construct, we grow and develop a vector containing our gene of interest, we infect the insect cells, we grow the virus, and with that we produce the proteins. So these are some of the insect cells which are widely used. Advantages are that it is integrated system with the DNA very stably, protein expressed in the native conformation, it can even express the secretory proteins, and no endotoxins, and less expensive than mammalian system. We are coming to the mammalian system also, but still there are some disadvantages. The disadvantages is that the N-linked glycosylation structure is different than the mammalian expression system, and the protease present in the cells sometimes degrade your protein of interest. But this system was widely used when we first encountered this particular pathogen, because mammalian system was not everywhere available, and during the COVID-19 pandemic, this particular expression system has been used to produce VLP-based vaccine, spike, RBD, ACE2, and many of the SARS-CoV-2 antibodies also has been produced through this system. So this is where what the system could help when we don't have the other advanced systems. In the mammalian expression system, what we do, we basically optimize our gene of interest nucleotide sequences that we called as a codon optimization. We transfect our cells, we do a pilot scale expression, we scale up the expression, and we purify the protein. These are the advantages. Here, what I have written, the authentic protein expression. It is something that gives us exactly what we want. Native environment for protein expression and folding, proper complex assembly, proper post-translation modification, reasonable level of expression. Because what you will learn in our next talk tomorrow, that how these cell lines has been modified and advanced even to achieve a very high level of expression. And these cell lines are endotoxin free. So this is the best system that what we feel right now we have. But when we also talk about the disadvantages, the first disadvantage is that it's a bit of costly system. We believe that in the future the cost may come down, but at this moment, people say that the longer expression time, but what we will learn tomorrow is that how we can reduce the expression time into a reasonable time frame. So this is a kind of a flow chart or a chart where you can see the advantages, challenges, and the most common applications. So here in the mammalian, you can see that with this system, we can produce the secretory protein, extracellular domain of the transmembrane protein, and this particular point is very important. In the further slide, you will see that how we can utilize this particular property in order to design some of the novel antigens and recombinant antibody expression and antibody fragment expression. So why do we need mammalian expression system? This is something that very important for us to understand. So when we talk about the eukaryotic and the viral protein, we know that there are many challenges which are associated. So we know that many of the conformations which we will deal is the dynamic and breathing conformation. These conformations keep on changing. So how can we design an antigen which will be a stable conformation that lead us to the isolation of the antibody? Another problem is that post-translation modification, that what we have already seen in the previous slide. Variable, metastable, open and close conformation, and shape-shifting pre and post conformation. So breathing and dynamic conformation has been shown in many of the viruses, but it's very common in case of influenza. Post-translation modification goes with the disulfide bound glycosylation. Open and closed structures has also been reported, and this is the RSV F protein pre and post conformation. As you can see, the pre-fusion conformation is a globular structure, and the post-fusion conformation is an elongated lollipop structure. So we need a different kind of engineering, different kind of understanding of the protein, protein structure, protein expression, and the knowledge of what we actually want to achieve from these antigen when we will move ahead in order to design an antigen. So next thing is the selection of target or antigen. How we could select a target, or how we should decide what antigen should we use. We all have known about the experience, we all have gone through the experience of coronavirus. We know that there exists a spike protein which is dimeric in conformation, and I am sure that some of you also must be aware about the RBD, which is a receptor binding domain. So starting from the coronavirus to spike protein to the receptor binding domain and to the receptor binding motif also, what should we use in order to isolate the antibodies? Because the S2 subunit, there are the antibodies which are known for the S2 subunit also. There are the antibodies which are known for the N-terminal, and also antibodies are known for the receptor binding domain. So it always depends upon us, what is our target, what we want to achieve when we want to decide what target should we use for the antibody isolation. I think some of you must be aware of that. All the antibodies which has been isolated using the receptor binding domain, there could be chances that they will be neutralizing antibody. Okay, so for you, it's a very important question to ask, what should we use? What will be our selection of the target? So I'm giving you 3 different examples, one of dengue, one of influenza, and another one of HIV. So here I'm just trying to show you some of the antibody derived questions that we ask, like if we want to isolate and if we want to have a unique antibody, what should be our choice of antigen? So the first slide over here is a dengue slide, and as I mentioned earlier, that our dengue envelope protein was dimer. And we know that in the absence of vaccine, the antibodies are something that is is widely needed. And we know that there exists is a concept of serospecific antibodies also, where antibodies targeted towards one serotype is not as effective in the case of other serotypes also. So here there are some reports where people have shown that the antibodies which are targeted at the dimeric interface of the 2 subunits, these particular antibodies are basically broadly neutralizing. They not only neutralize all the 4 serotypes of dengue, but they also neutralizes very efficiently Zika also. So how we could achieve this particular conformation? So in order to achieve a dengue dimer conformation in a soluble protein conformation, what we have done and what you could be also done is that we have introduced a cysteine residue. And we know that if 2 cysteines are very close to each other, they will form a disulfide bond. So as you can see that all the red highlighted marked circle here is the cysteine residue. These are at the interface. And in this interface, we introduce the cysteine residues and they form the disulfide-disulfide bond. By forming this bond, they basically cross-link the 2 subunits together. Not only this technique has been used in order to generate dimer, but this technique has also been used successfully in order to generate a stable trimers also. I will be talking about influenza HA. We know that every year we need a new influenza vaccine because influenza mutates so much that the antibody that body generates after first season immunization is no longer effective in the next season. Why? Because our HA, which is the main target for the production of antibody, keeps on mutating. There are some of the broadly neutralizing antibodies which are known for the influenza, but the problem is that they are not that potent. So there are some of the literatures that shows that there are some antibodies available, but the problem with these particular antibodies were that they were not targeting towards the external surface of the influenza. So how should we isolate these antibodies? How should we design our antigen? So here we had 2 conformations, one which was the closed conformation of HA as a timer, and one what we wanted is a properly folded open conformation. So in order to design that, what we did, we designed a HA trimer, we introduced a protease which is shown, protease cleavage site which is shown by a simple single line over here. Then we added a trimerization domain and a histone. Because for every protein, in order to attain a proper conformation and proper folding, it's also necessary that if a protein is a trimer, it should fold as a trimer. So once the protein folds as a trimer, expresses a trimer, then we cleave the trimerization domain, and what we get a stabilized monomer. But those who have studied the protein, we also know that when 2 protomers come to each other, they forms a trimer, dimer or tetramer only because of that, because the the amino acids which are present next to each other, they have a tendency to interact. So even if you will remove one of the subunit, they will have a tendency to fall apart. What we need to very carefully look into the sequence and to identify, are we having any of the sequence which will make our protein insoluble or it will make it to fall apart. So with that, some of the analysis, we identified couple of the residues, we mutated those residues. And what we finally achieve is a conformation that could also be able to isolate and that could also be able to help us to purify the monoclonal antibody, which not only targeted towards the external surface but also towards the internal core. Another target that I will be talking today is a HIV envelope protein. We know that still now we have no vaccines for HIV. Antibodies are something that is very widely used. There are many antibodies which are known. But the problem with the HIV is the diversity. The HIV is so diverse that within a one region, within a one clade, you will find thousands of the subclades. So that makes things very complicated. From one region to another region, from one clade to subclade, the sequence completely keep on changing. So in order to understand that, people have done design a construct that we called as a ectodomain, which is a surface expressed, the region which is present above the surface, and a success has been reached where people was successfully expressed up to 664th amino acid. But the HIV envelope protein has a very long C-terminal domain, which is inside the cytoplasm. But the literature also shows that there are two antibodies which are known, which are targeted towards the cytoplasmic tail of the protein. Now here the question comes, when this particular region is inside the cytoplasm, how the antibodies are actually exist. So there are couple of the theories and couple of the hypotheses are given that shows that this particular region at some point of infections also comes out of the surface. That make this region accessible to the B cell so that we have these antibodies. So if you want to use this particular concept into your design, how you will do that? So what we did, this is something we believe was actually the conformation. Either we had this particular conformation or we had this particular conformation where this surface was exposed at the surface. What we did, we deleted the transmembrane domain. We added a linker. We did some of the stabilization mutation also, as I explained earlier, based on the sequence. And with this methodology, what earlier was available only up to 664th amino acid or to 680 amino acid, we attained a conformation where we were very successfully expressed the construct up to 754th amino acid in a soluble conformation. So these kind of antigens we actually need if we want to isolate something unique antibody which we will be actually needing for our therapeutic purposes. So what takes us to the designing, that what I called it as a structure function based designing. If we know or if we understand the structure, if we have the knowledge of the structure, or through the bioinformatics analysis, if we can model the structure, if we can analyze the structure, what we go, we first predict the domain or the folded conformation of the antigen. Second, we further looked into the transmembrane domain, signal peptides. We looked into the post-translation modifications, sequence analysis, disulfide bond, salt bridges, so that when you are designing a construct, you should not do this kind of deletion that your conformation get distorted. Structural stabilization mutation. I showed you how we introduced couple of the mutation in order to stabilize the conformation. Then we looked for the appropriate expression system. Does it actually need glycosylation disulfide bond formation? If so, we move into that expression system. If not, we go back into the easier or the cheaper systems. Then codon, construct designing, codon optimization, synthetic gene synthesis, expression system. I'm talking about the mammalian, so I have written mammalian here, protein purification, then extensive functional characterization. Whatever you have designed is actually serving the purpose that what we wanted, and that leads to our antigen and target designing and development. So where we started, in addition to that, in order to achieve a near native conformation, we do some of the engineering, and this engineering also involve unique epitope-based heterologous protein structure function-based designing that I explained, mammalian expression system and molecular clamps. Here with this methodology, we have developed some of the methodology where we can also express only one epitope if we want. Though I explained you earlier, it's not an ideal condition, but if we come up to that, we can do that also. We can use different molecular camps if we want to express protein into a nanoparticle. So with that, what is an antigen? I am introducing another term, immunogen also, and with that, at present, we are working on all of these pathogens and vectors. So at that, I would like to end my talk, and I would like to acknowledge my lab, my collaborators, and different resources, platforms which help to do this work. So with the tagline that monoclonal antibodies could be an emerging era in the biological, so I would like to end here. Thank you. Questions?
Thank you, Dr. Tripti. It was a wonderful presentation. You gave a very good overview of the protein production systems and how important it is in the monoclonal antibody generation. So, any questions from the audience? We expect a lot of questions. It was a wonderful presentation.
So, thank you, Tripti. I feel you enlightened a right topic, because whatever we invest, we will get. I think that's the right thing. And in antibodies also, it's my experience, I also feel that when we have a right antigen and a right combination of adjuvants, that also makes a lot of difference. How we are presenting, that also matters a lot. And
it's totally depend upon how we are presenting our epitope into the right conformation. If we have the epitope into the right conformation, we will definitely get the antibodies what we actually need.
Absolutely right. Yeah. Thank you. Yeah.
Any questions, students? Yeah, please, backs.
Hi ma'am, I'm Lakshmi from Indian Veterinary Research Institute.
Yeah.
Ma'am, can you please elaborate the principle behind the baculovirus expression system?
So the baculovirus expression system is a two plasmid expression system where we have a virus-based system and we had a helper plasmid. We clone our gene into one of the normal cloning vector. We transfect into along with one of the helper plasmid. What happens? Our gene of interest get transferred into the viral genome, and that basically start producing our gene of interest in terms of on the phase of a virus. So basically, we produce a virus. We use this virus to infect our insect cells. Once they get infected, like a normal, as we infect our cells with the virus, they start producing the proteins. So that comes into the cell if we have not added any secretory signals. If you have added any secretory signals, your proteins come into the supernatant and we purify the protein out of that. Anybody?
Yeah, good afternoon, ma'am. Myself, Anush from JSS College of Pharmacy, Ooty. I want to know about the role of adjuvant with antigen and how we select adjuvant for a particular antigen.
Sorry, I cannot understand. What you said?
Yeah, I want to know about the role of adjuvant and how we select a particular adjuvant for a particular antigen.
Can you come ahead? You are asking for the adjuvant?
Yes, ma'am.
So when we go for the immunization, we basically use the adjuvant in order to help our immune system for the generation of better immune response. So basically adjuvants, what they do, they basically enhance, they make the release of the adjuvant slow, and they basically help into the generation of T cell response. So when we talk about the protection, it's not only the antibody, the humoral response, our T cell response, which is the cellular response, that also very important. The antibodies, where they neutralize the pathogens or the virus or anything, but the T cells are the something that helps to eliminate that particular pathogen from the system. So these are the two basic side by side, you can say, the system arms that runs together. So for the generation of the cellular response, adjuvants are very important.
And one more question is this, like we are doing the genetic engineering for the antigens, we are taking the particular gene of interest for particular antigen, like we are taking the NS1 and domain third of dengue. Like this we can do for antibodies also, like particular epitopes of antibody, and we will go for that.
So see, antibodies get generated in response to the epitope. Antibodies has paratopes.
Yeah.
So antibody doesn't have the epitopes. You can generate antibody response in terms of or in against a epitope. So you can have one of the epitope, you can design your construct so that you can express an epitope and your antibody will be directed towards that epitope.
Okay.
Okay. So when we call about the Y-shaped structure of the antibody, basically the bottom of the Y is the Fc and the two edge are the Fab. So both Fab has the property to bind to the epitopes. So if we are talking about the one antigen, they can bind to the similar epitope. But we can go into the antibody engineering. We can design bispecific, trispecific, and then tetraspecific antibodies, where we can make these antibodies to bind to the multiple epitope at the same time. So this is further a step of antibody designing, where can you design multi-specific antibodies also.
Thank you, ma'am.
Okay, thank you.
Any more questions? So maybe we can continue the discussion. Yeah, we have one more question.
Ma'am, you have talked about the Pseudomonas aeruginosa, which is associated with the catheter associated UTI. Does this antibody works for this case also?
Which one?
UTI.
Yeah, so
catheter
basically this is under the clinical trial. People have isolated the antibodies and they are in the different phase of the clinical trial for the actual therapeutic use.
Thank you.
So thank you, Trupti, for a wonderful presentation. So we will have the next talk by Professor Deepak T. Nair.
So we are welcoming you to the dais. Professor Deepak T. Nair is a professor at Regional Center for Biotechnology, Faridabad, Haryana. He did his PhD from the National Institute of Immunology in New Delhi and postdoc from Mount Sinai Medical Center, USA. He was awarded the National Bioscience Award in 2014 and the Shanti Swarup Bhatnagar Award in 2017, which is considered as the Indian Nobel Prize. He is a member of various DST, DBT and ICMR committees. Currently, his laboratory at the RCB studies on the enzymes from pathogens that replicate or repair the genome and their role in the onset of resistance towards the therapeutic or prophylactic agents. He is also interested in the design of therapeutic antibodies that are resistant to the appearance of variants that escape neutralization. We are happy to have you here, sir.
We have to put this charger on. Showing up. Okay, I'll share. not having. Will this screen also be shared? No, this is just the PowerPoint. What if I want to show something else?
Can you screen share? You can open that file later.
Okay. Okay. All right. Sorry for the delay. Thank you for the introduction, and I would like to thank Rajesh and Dr. Ishrukumar for inviting me here. I actually, I have some sort of background in antibodies, but only recently, thanks to due to Rajesh, that I have come back to antibody structures. So I did do my PhD on antibody peptide complexes, and then after some 20 years later, Rajesh popped up in my office, and we have solved another antibody structure. So I'll just give a brief overview of some basics about antibody structures, especially the variable region, and then tell you a little bit about the work that I and Rajesh, along with Devinder and Chandresh and Tripti, have done on the anti-SARS coronavirus antibody. Okay. So this is what your textbooks, especially the very, very wonderful textbook by Janeway and Travers, Immunobiology. This is the kind of cartoon picture that you will see. There is a Y-shaped molecule. There's on the Y, in the arms of the Y, half of it is light chain, half of it is heavy chain. And there are two arms to the Y. And then you have another part which is completely heavy chain, which we call the FC region. And this is the more, it's a better representation of what the actual structure looks like. You have a variable heavy this laser doesn't show up very nicely here. So this is the you see a variable heavy domain, and then there's a variable light domain, and then there is a Cl domain, CH1 domain, and in the FC region you have a CH2 and CH3 domains. And there are disulfide bonds present between the two heavy chains, and there's a hinge region between the stem and the arms of the Y. And the binding sites are located over here. So the actual structure looks like this. This is one arm which will bind to the antigen, this is the other arm which will bind to the antigen, and this is the FC region. And each of these domains that you see here is representative of what is known as the immunoglobulin fold. So immunoglobulin fold is generally defined as what is known as a beta sandwich, in the sense that you have one beta sheet and then you have another beta sheet, and they are sitting on top of each other and joined together on the inside by disulfide bonds. So this is very characteristic of a immunoglobulin fold, and each of these domains that you see here will be a representative of the immunoglobulin fold. So why is this immunoglobulin fold so helpful in terms of antibodies? Now when people started getting the first sequences of antibodies and they started comparing the sequences, they saw that in the variable region there were 3 regions which had very different sequences compared to the rest of the protein, and these were defined as the high complementarity determining regions. In both heavy and light chain you saw that the variability increased in 3 regions, and then whereas the rest of the protein was relatively, the sequence was relatively similar. So how does this translate to structure is what people try to figure out. And for that, what they did was they took the whole antibody and using papain, they cleaved the whole antibody into 2 Fab fragments and 1 FC fragment, and then they could crystallize this Fab fragment in complex with antigen alone as well as in complex with antigen and solve multiple structures. And so lots of antibody antigen Fab fragments of antibodies in either alone or in complex with antigen have been determined, and this has not only told us a lot about how antibodies function but also about how proteins bind to each other or bind to other macromolecules. So from a general principles of macromolecular binding, also this kind of studies have been a big gold mine which has led to very a deep and very fundamental understanding of how macromolecules interact. This is what the structure looks like. So this is Fab bound to on your left side is a Fab bound to peptide antigen, and on the right side is a Fab molecule bound to a protein antigen. And not only so, what happens, the way the beta sandwiches are arranged in space, you have these 3 loops which are shown here as L1, L2, L3 and H1, H2, H3, which are arranged in space which are close to each other. And these 6 loops, 3 from light chains, 3 from heavy chain, together form the antigen binding site or the paratope. Okay. And the composition, the length as well as the composition of these hypervariable loops can change, and that is why the surface which they form will keep changing for different antibodies. So what the immunoglobulin fold allows is you bring 2 immunoglobulin folds close to each other, and then the loops within those immunoglobulin folds will come close to each other, and you generate a surface which you now can change by just changing the length and composition of the hypervariable loops. So that way you can generate multiple binding sites for multiple different kinds of antigens. And this is the peptide antigen sitting in the antigen binding site. You can see that residues from all the hypervariable loops come close to the peptide antigen, and residues from all these loops will be involved in stabilizing this antigen in the antibody in the antigen binding site. Now how does the antigen sit here? And that happens primarily through non-covalent interactions. So this is what you see, that with this nice design of 6 loops coming together in space, you can generate binding sites which can bind to a variety of antigens. You can have binding sites which bind to small haptens like organic molecules. So there will be a small hole like groove, small cavity on the antigen binding site, and the organic molecule can sit there. You can have peptides binding, you can have large proteins binding, and you can have entities, macromolecular assemblies which are actually much bigger than the antibody itself, can also be bound by the antibody. For example, viruses can be bound by the antibody, and here you see that the shape that is presented by the antigen binding site, which is defined by the length and composition of the hypervariable loops, can be different, and you can bind antigens of different shape as well as size with very high specificity. And this specificity arises primarily due to these non-covalent interactions, which include electrostatic forces, hydrogen bonds, van der Waals forces, and hydrophobic forces, which is hydrophobic regions coming together in coming close together. And this kind of interactions are very important because in an aqueous milieu, these interactions are actually very, very strong. And since all, in case of biology, all interactions are happening in some sort of water solution, these hydrophobic forces play a very important role in stabilizing interactions between macromolecules. So those 6 hypervariable loops will present residues which will form non-covalent interactions with your target antigen, and the array of non-covalent interactions that they form will be very specific for that antigen. Both the nature of the interaction as well as its position in space will be very specific for that antigen, and that is how you get high specificity of antigen-antibody recognition. So you have read in your textbooks about, you need, from an immune system point of view, there has to be antibody which can bind to any and every antigen that will pop up inside our body. And so how do you generate all this entire range of antibodies? And what you can see in your textbooks is that there is somatic recombination which brings together different antibody segments together in space. And what happens is you have these V and J segments in case of light chain and VDJ segments in case of heavy chain. And due to somatic recombination, these genetic segments will come together in space. But the region which corresponds to the junctions between these segments, in terms of antibody structure, they correspond to the hypervariable loops. Okay, so that is how you can generate different kinds of hypervariable loops through this VDJ recombination. The other thing that one should always remember is that when these gene segments are joined, at the junctions you have these nucleotide additions, P and N nucleotides, which are appearing due to the nature of due to the process through which these segments are joined. And so this leads to random addition of nucleotides at the joining segments, especially true for the DJ joining. And that is why this TDT enzyme will randomly add nucleotides during this joining process. So you have certain fixed number of gene segments, and that can give you a fixed number of combinations regarding the antigen binding site. But due to this random addition of nucleotides, you can generate very different length as well as composition of the hypervariable loops, and that adds a lot to the diversity that you see in the antigen binding sites. And that is why, since this addition of nucleotides happens more at the junctions, so that is why you can see that the length and composition of these junctions, especially the DJ and the VJ junctions, they correspond to the L3 and H3 loops in the antibody structure. And that is why you see maximum variability in the CDR3 of the especially CDR-H3 will show the highest amount of diversity both in terms of length and composition. And that is what allows you to generate a lot of, so all these together allow you to generate a number of different binding sites which can specifically recognize different kinds of antigens. But this is still not enough. So this is all there in your textbooks. But what is now becoming clear is that there is a the way antigens bind to the antibody, the same antigen, the different antigens can bind to the same antibody at different locations in the paratope, in the antigen binding site. And this is especially true in the germline antibody. In the germline antibodies, you can have this situation where different antigens can bind to different locations on the antigen binding site. And then once that primary engagement happens with low affinity, then you will have affinity maturation and somatic hypermutation, which will improve the affinity for that antigen antibody engagement. So this is this new kind of structural strategies to enhance diversity at the germline level. This has come up in the last 10-15 years, and Dr. Dinkar Salunke at ICGB has been one of the main contributors to this discovery. So these were a little bit about the basics of how antibody structures are, the design of antibodies, and how the arrangement of hypervariable loops allows antibodies to bind different kinds of antigens. So now I'll talk about some of our work on P4A2 Fab in complex with spike RBD from SARS coronavirus 2. So you know all this, this is the SARS coronavirus, and there is a spike protein on the surface of the coronavirus. And this spike protein actually forms a trimer, and this is the domain organization of the spike protein. There is a receptor binding domain on the spike protein which binds to the ACE2 receptor of humans, and there is a region on the receptor binding domain known as a receptor binding motif which contributes to the interactions between the spike protein and the ACE2 protein. So Rajesh, who's now at IAV but was at THSTA before, he and Chandresh had they had using mouse hybridoma technology, they were able to find this P4A2, they were able to generate this P4A2 monoclonal antibody which can recognize the spike receptor binding domain of spike protein with high affinity, and they saw that it can neutralize different SARS coronavirus 2 variant of concerns. Alpha, beta, kappa, delta, all could be neutralized, and it can bind to the spike RBD from these viruses with very high affinity. So these are, these are very high affinities. So he wanted to solve the structure, so he gave the proteins to us, and what Naveen Narayanan, who's actually from Kerala, he's from Thrissur, he was the student who worked on it. So he mixed the two proteins together, the P4A2 Fab and the spike RBD protein, and he carried out a gel filtration experiment, and he was able to get a clear peak for the Fab RBD complex, so which was concentrated, and he set up crystallization experiments with the complex. So in this crystallization experiments, you basically carry out hanging drop experiments where the protein is in the drop that is shown here, and there is a reservoir with different kinds of precipitants, and the whole thing is sealed. So basically the drop undergoes slow drying over time, and when this happens, it forms a supersaturated solution of the protein, and that can either form precipitate in most cases or crystal in few cases. So that is how he was able to find conditions which give rise to crystals which look like this, and the crystallization conditions that he found involved magnesium formate and 10% PEG 5K-MME. So this is these crystals were actually first tested at our home X-ray source in RCB, and they showed diffraction to up to 4 angstroms. and the home source was also used to standardize the cryoprotectant for these crystals, which was 20% glycerol. So cryoprotection is necessary when you're doing crystallography because you're exposing these crystals to high intensity X-ray radiation, and that can damage the crystals. But if you freeze the crystals and collect data at 100 Kelvin, then the damage is minimal. So this is, the data was collected at ID30A1 beamline at the European Synchrotron Radiation Facility, which is located in France. And this is what a representative diffraction pattern looks like. So the crystals, we could collect data till 3 angstroms, and these are what are known as the data collection statistics. And the structure was determined by molecular replacement method using the structure of another fab in complex with RBD. So that fab will have a different sequence, but we could the sequence was similar enough so that we could solve the structure by molecular replacement. Then the sequence of that fab was slowly changed to the P42 fab, and iteratively you carry out what is known as crystallographic refinement till everything is okay, till the refinement converges. So these are the final refinement statistics, and the structure is now deposited in PDB with the PDB code 7WVL. So this is what the structure looks like. The spike RBD is shown in magenta. Orange is the heavy chain and cyan is the light chain. And this is a close-up of the antigen binding site, which shows all the residues which are engaged in interactions. So this is very busy, but here is a snapshot of the key interactions which are holding the antigen, which help the antigen bind to the antibody. So on the left and right hand sides and the top middle, you can see what are known as hydrogen bonds between residues of the spike RBD and the antibody. But one of the key interactions in this is this F486 residue, which is a phenylalanine residue. The aromatic ring of the phenylalanine residue is present in a hydrophobic cavity formed on the antigen binding site. And this is, you can see this here, there is where F486 is written, there is a cavity, and into that the aromatic ring of the phenylalanine residue is present. So this, like I said before, this is a hydrophobic interaction which would be very strong because all antigen antibody interactions are going to happen in water, in an aqueous solution. Now, so this is the structure of the antibody bound to spike RBD, and this is the structure of ACE2 bound to spike RBD. And the residues that are shown here, leucine 455, phenylalanine 456, phenylalanine 486, asparagine 487, which you can see come close to the ACE2 receptor residues, are also very important for binding to the P4A2 fab. So now you can immediately guess what is going to happen. This is the P4A2 spike RBD structure. If you overlay the spike RBD structure from the complex with ACE2 onto this, you can see that there is going to be a steric clash between the fab molecule and the ACE2 molecule. So if P4A2 monoclonal antibody is bound to spike protein, there is no way that the spike protein can now bind to the ACE2, and that is why the infection, the viral titers will go down drastically in the presence of P4A2. So the spike trimer is known to exist in two forms, where the receptor binding domain can be in the up conformation or in the down conformation, and the nature of the P4A2 engagement with the spike RBD is such that whether it is up or down, it can still bind to the RBD. It's only when the RBD is in the up conformation that it can bind to the ACE2 receptor. So, and the affinity of P4A2 monoclonal antibody is about 1000 to 10,000 times more than the affinity of ACE2 for spike RBD. So that because of this, there is, if there is P4A2 monoclonal antibody present, there is very little chance of spike being able to bind to the ACE2 receptor. So with this structure, we could also model, using this structure as a template, we could model what, what the binding of P4A2 Fab could be to the other variants of concern. So in case of beta, there are 2 mutations, and we see that 3 mutations, that none of these mutations will perturb the binding. In case of gamma, there are 3 mutations, they will also not perturb the binding. In case of delta, there are 2 mutations that will also not affect the binding. These are all mutations in residues which are not interacting with the P4A2 Fab. So that is why they will not affect binding. And in case of kappa also. In case of the BA.1 Omicron strain, there are about 15 mutations. But again, all these mutations are residues which do not interact with the P4A2 Fab. So the hypothesis was that P4A2 will also be able to neutralize Omicron BA. And that is exactly what Rajesh, meaning through experiments done by Rajesh and Guru Prasad in THSTA, that was shown that the virus, Omicron virus gets neutralized, as well as the spike RBD binds to, from Omicron BA.1 binds to the P4A2 with very high affinity. This is a comparison of the binding sites of different antibodies that are in the literature to the spike RBD, and you can see that there is some overlap between P4A2 and 87G7 and COV2 2196 and a few others. But this interaction that we see, that the phenylalanine is buried deep in a hydrophobic cavity on the surface of the antibody, that is very unique. That is unique for the P4A2 antibody, and that is probably by one of the major reasons why it is able to neutralize, it is able to bind with such high affinity and neutralize different VOCs. So this is the summary from this part of the talk, that P4A2 provides protection against tested VOCs, and the humanized version of this P4A2 antibody might be a very effective therapeutic against different VOCs. So this paper is in process. Now to wind up, I just wanted to give you a quick insight on what is the, if you solve structures of antibodies, alone or in complex with antigen, you get to know what is at the atomic level, how does this antibody bind so specifically to the target antigen. So what is the use of this information from an application point of view? Now, the structural information can be used to engineer the antibody to have desired properties. And one of the desired properties, like I mentioned, is that humanization of murine antibodies. And generally, there are other strategies, but some of the strategies that, meaning one of the strategies that people use is known as CDR grafting. So you have a murine antibody which binds to a target antigen, but you want to humanize it. So you take the CDRs from that murine antibody and you stitch it to a scaffold of a human antibody. So in making the decision as to which human antibody scaffold to choose, if you have structural information, then that becomes a lot easier. And also, if you have structure of the murine antibody in complex with antigen, you now know in the scaffold of the human antibody what are the residues outside the paratope that you might have to mutate so that you generate optimal affinity. And meaning Tripti, during the discussion with Tripti, this issue of bispecific antibodies had popped up. So when you try to design bispecific antibodies, which like Rajesh said, you have two arms of the Y, usually both arms of the Y will bind to the same antigen. But in a bispecific antibody, one will bind to one antigen, then the other will bind to another antigen. And you can generate this by engineering, stitching different variable regions to to one antibody. So here is a schematic of how this can be done. You see these D3, D4, D5, D6. These can be different variable regions which have different specificities which can be stitched together. In a bispecific antibody, you will only have D3, D4. So, but if you want to generate more specificities, like you want to have trimeric or tetra specific antibodies, you can have D5 and D6 also. And all this engineering becomes easier if you have the structures or if you use structural information which is available. The structural information can also be used to improve solubility and stability of antibodies, and like there was a proprietary monoclonal antibody from Pfizer, and that the solutions of that antibody were very, very viscous. So, but based on structure modeling and structural information, they were able to find out few residues which they could mutate. So they mutated the 45th residue, which was valine, to lysine, and that reduced the viscosity substantially. And the stability of the antibody molecule also can be increased by, based on the structure, you can bring in residues which form intramolecular interactions, and that will increase the stability of the antibody, so that even if the temperature is a little higher, the antibody properties, binding properties remain intact. And this becomes important, you don't have to worry about cold chain and things like that. And the most obvious thing that one can think of when you have structure is synthetic affinity maturation, where you can identify residues that can be mutated to different residues to bring in new interactions, new non-covalent interactions between the antigen and antibody, and that will enhance the affinity of the antigen for the antibody. And one example is this trastuzumab, which is antibody directed against human epidermal growth factor receptor. So there is one mutation, 98 residue aspartic acid to tryptophan, that led to 3-fold affinity improvement. So like that you can carry out synthetic affinity maturation of antibodies to improve affinity and specificity. And for P4A2 also, this approach will have to be employed, because you have these new Omicron lineages where that 486 phenylalanine is mutated to valine. And not only to valine, now you have all these lineages emerging. So this is from a tweet from Professor Adrian Easterman from Australia in October. Wherever those arrows are pointed out, I see F486 is mutated to proline, to valine, to serine, to isoleucine. So all these new variants, and this is like in the last 3 months or so, these new variants are emerging, and the chances are very high that P4A2 will have less affinity for these variants, because that phenylalanine interaction is reduced, that hydrophobic rate. But we can look at the structure and now design new residues and mutate residues at key positions and try to bring back the affinity. One of the important things that should be mentioned here is that when we saw that residues which interact of spike RBD which interact with P4A2 Fab are important for interacting with ACE2. So, the chances, so if there is any mutation in those residues, then the affinity for ACE2 should also go down. And that does actually happen. When you have F486V, the affinity of spike RBD for ACE2 goes down. But then there is another mutation, R493Q. Arginine 493 is mutated to glutamine. And that brings back the affinity to a level that now the infection can set in. So what I am trying to tell is, even if we have antibodies, it is going, especially against RNA viruses, we have to be constantly evolving those antibodies, because the virus is also going to constantly evolve. With that, I'll end. Naveen did all the work that I showed, and these are the other people in the lab. Funding was from Department of Biotechnology, RCB, and we use X-rays for our work, which is from the home source at RCB and also from ESRF. And before I finish, I just want to tell you about these RCB programs that are there that are useful for all the young people sitting here. One is the ESRF Access Program. So if any of you are into structural biology, macromolecular crystallography, and you want access to synchrotron, there is a program funded by DBT which will help you do your experiment. There is Advanced Technology Platform Center in RCB, where there are 6 different facilities which you can send samples, or you can come with your samples. You pay some charges and you can do experiments. Then this is the Indian Biological Data Center, which is going to be a repository for all life science data from India. If you are generating sequence data, please try to deposit it in the IBDC. There is this Indian Nucleotide Data Archive. Please deposit it there. Once you deposit your sequence in INDA, automatically you will get an accession for GenBank and EBI access databases. And this is a new thing which is coming up, which is a Bioinformatics Center for Structure-Based Computational Drug Discovery. Within a year, anybody who wants to carry out computational drug discovery can come and use the resources that are present in this facility. Thank you.
Thank you, Professor Deepak. Any questions? Yeah.
Invariably, you were saying about if the structure is available.
What?
Many times you are saying that if the structure is available,
yeah.
For the antibody engineering, for many type of things. So all that means that the structure availability is only through X-ray crystallography?
Now you can solve structures by different ways. You have NMR, you can get structures of small antigens bound to FV region through NMR also. Macromolecular structure has, but now you also have cryo-EM. So many of these recent spike trimer with Fab complex structures have come through cryo-EM.
Okay. So what about, like, for example, the identification of D98W mutations? So how do you determine that?
You can either get experimental structures and use that, or if there are no experimental structures, then you can try to use modeling, computational modeling, to get some structural insight into how the antibody looks like.
Okay, actually I was asking a question thinking that you may say that mass spectrometry might be used, because I'm a mass spectrometry person. So does mass spectrometry has any use?
Mass spec can tell you whether, with a cross-linker, can tell you which residues are close in space. If you do peptide fingerprinting of cross-linked samples of antibody and antigen, that can give you an idea which residues are close in space.
What about the primary sequences about the hypervariable regions, etc., in the therapeutic antibodies? Is there any future for mass spectrometry?
If the sequence information comes from genome sequences, which is always better. But there is some amount of evidence that sometimes post-translational modifications
glycosylation.
play an important role. You know, there are post-translational modifications in the FC region which are very important and have a big influence of different properties of the antibody. But there is some evidence that, you know, there can be post-translational modifications close to the paratope also which influence antigen binding. So in those cases, mass spec will be very, very useful to figure out what kind of post-translational modifications they are.
That is nice to hear.
Thank you. Any more questions? Yeah.
Nice talk, sir. My question is, like, somewhere you mentioned that P482 Fab has more affinity to bind with the ACE2 receptor rather than the spike RBD. My question is, like, is it through the structural data, like finding your team the critical residues that interacts between these two, you understand the affinity, or like have you done any other experiment like functional characterization?
Has done the experiment and they have seen that using biolayer interferometry, that you know, it has, I think, 10,000 times more affinity.
Okay, so it's not only through the structural
experimental evidence.
Okay, okay, thank you. One more doubt I have is like you mentioned the cryoprotection of the crystallites. So is it possible with every crystal, or like?
No, you have to do it for every crystal.
Okay.
Because the intensity of the X-rays that, you know, we are exposing these crystals to, they are like, you know, like very, very high compared to your medical X-rays and all. These are very, very high, and they lead to, you know, they can cause chemical changes in the proteins in the crystals, and after some time proteins, the crystals can stop diffracting. So to avoid all these problems, you collect data at very low temperature. You have to freeze the crystals and collect them at low temperature. So that is why cryoprotection is very important, because if you freeze anything, you will immediately get ice formed. Ice is actually a crystal of water. You can't have a crystal within a crystal. Your protein crystal will go for a toss if ice forms. So the aim of cryoprotection, both in case of macromolecular crystallography as well as in case of cryo-EM now, is to get water to form, instead of ice, it should form an amorphous solid, so that it does not interfere with what is going to happen.
Okay, so it doesn't have, like, it doesn't affect the structure or like the stability of the. Okay, thanks.
So, thank you, Professor Deepak.
Can remove this.
We are kindly requesting Dr. E. Srikumar to hand over the token of appreciation to Professor Deepak T. Nair, sir. We are requesting Tripti ma'am to be on stage to receive the facilitation from Dr. E. Srikumar ma'am. We are requesting Dr. Mohanan Valivittal to hand over a token of appreciation to Gopalakrishnan sir for chairing the session.
So we are moving on to the next technical session, which will be chaired by Dr. Anusmrita Lahun and Dr. Ashwati Raj. Dr. Anusmrita Lahun received her PhD from National Institute of Virology, Pune and Savitribai Phule, Pune University in 2014. She did her postdoctoral studies in the Department of Virology and Immunology at Baylor College of Medicine, US. She joined the Institute of Advanced Virology as Scientist C in 2021 and is working on developing the nucleic acid-based vaccines for Zika and Chikungunya viruses. Dr. Ashwati Raj graduated from Medical Laboratory Technology from Medical College, Trivandrum, and did her PhD in Virology from Manipal Institute of Virology. She was the recipient of INSPIRE Fellowship and International Mobility Grant for doctoral students in 2014. She joined the Institute of Advanced Virology as Scientist C in 2021. Her research interests are public health aspects of virology, emerging and re-emerging viral diseases, diagnostic virology, and epidemiology and diseases. So we welcome you, ma'am, and I kindly request the chairs to make the next announcement.
Good afternoon, everyone. On behalf of IAV, we are welcoming Romsa Kumar for this first technical talk. She will be going to deliver on a talk on single cell analysis system, a robust microwave-based single cell partitioning system for high dimension biology research. Please, I'd like to welcome her. So over to you. Welcome, Dr. Romsa.
Dr. Romsa Kumar is working as an application scientist for single-cell multi-omics in BD Biosciences, India. Before joining BD, she worked as a scientist in the Department of Biochemistry, AIIMS, New Delhi. She received her PhD from AIIMS. She has more than 13 years of research experience in the field of immunology with expertise in HIV pathogenesis and chemokine receptor signaling. Dr. Kumar has an excellent experience in single-cell multi-omics, cell sorting, and multicolor flow cytometry. We welcome you, ma'am.
Thank you so much. First of all, I would like to thank Dr. Rajesh and Director Sir for giving us an opportunity to talk about this technology. So before going and presenting my slides, I would like to say that why you want to use this technology, what is the advantage and why should I go? So from the morning talk you are listening about all these monoclonal antibodies, how they play a role, what is their, how to characterize them, how to see the immune response. So today I'm going to talk about this single cell technology, single cell multi-omics technology, where you can, with the help of this technology, you can have fully characterize your T cell receptor as well as B cell receptor at a single cell level. Not only you are going to characterize your full length VDJ part, as well as you can have their transcriptome and their protein part also. So that is why we are saying it is a single cell multi-omics technology. So the question is, why do we need to know about the single cell? First, the answer is to resolve cellular heterogeneity. As we know that mRNA and protein are not always correlated. So I'll go in detail about the VDJ part and TCR, PCR, but first, for all the student in the audience, you have to understand what is the advantage, why we wanted to identify the cellular heterogeneity and from where it occurs. So during the time of the development, due to the post-translational modification and other things, there is a changes. So that will a single cell become a different kind of a cell. You can recall your 12th where this gastrula, bastula from single cell to a full different kind of a system. So, and in case of any pathological situation also, there is a change. So what kind of a change? Like recently we were all seeing that there is a COVID was there, and some people are having this asymptomatic COVID and some are having symptomatic. What was the change? Where there is an immune response and how it is leading to the changes and then all. So to resolve this, we have to do this single cell. So what is our general way, means normal way of doing this single cell analysis, how we do it in our lab? Either if we wanted to identify protein, we check the protein expression by microscopy, by mass spec, and then flow cytometry, what kind of immune system. And then we check the qPCR and the bulk PCR analysis through the RNA-seq, and then we do this. So the BD has come with a new machine. Just a little bit overview of this machine, which is known as BD Rhapsody, which is a microwell-based partitioning system to characterize the single cell. And it's a single cell handler. It comes in a 2 configuration, that is a scanner, this is the scanner, and this is the express system. Can I have the pointer? Okay. So what is our offering is like, first, to do an experiment, you should have an instrument. So the instrument is the Rhapsody scanner, and the rest other things are these reagent. So this reagent is this whole, if I'm saying it's a multi-omics technology. So multi-omics where we can see both gene as well as your protein. So gene part is taken care by this whole transcriptome amplification kit as well as the targeted RNA-seq. And the protein part is taken care by this AbSeq, which is a protein expression part. The protein expression analysis, why we are saying is AbSeq, because this antibody structure, which you use it for your Western blot or float, the same clone will be there, but this clone is tagged with the poly A tail. So that is why we are, it's an oligo-based antibody, so that is why we are saying is AbSeq. And the overall, the readout of this experiment is through sequencing only. So next generation sequencing technology through any of the NextSeq or NovaSeq, any of the Illumina sequences. So other than this, we have this VDJ kit where you can fully characterize your T cell receptor as well as B cell receptor. And we have this sample multiplexing where you can pool different samples together and then you can analyze. So this is all about the kit, and with the help of this kit, you can make a library which is ready to load on the sequencer. And then we have a pipeline where you can check what could be, with the help of the pipeline, when you load your FASTQ file, it will give you the CSV format files, and then you can export into the either your SIGIC software or any of the third-party software, and you can further analyze your T cell receptor, B cell receptor, your transcriptome, as well as your immune profiling also. So this is about what is the Rhapsody scanner and express system. So what is the advantage of having a scanner is that it gives me the visual workflow. Visual workflow means it will tell me how many cells I have loaded, what is the viability, what is the multiplet rate. So that is the advantage of having Rhapsody scanner. And the express system where you can, it's a mechanical system where you have to load the cells and you have to do all this practical work like lysis, loading and all. So this is the overall view how we work. First we load the cells. So as I said, our system is a microwell based system. So we have a cartridge where there are more than 2 lakh microwells are there. In that we load the cells. Then we load the bead and then we lyse the cells so all the RNA will come out. And then we'll have this magnetic bead which is having poly-DT tail. And then all the RNA which is having poly A tail will bind to this magnetic bead, then we can retrieve this bead and then we can synthesize the DNA and analyze the data. Now the question comes about the AbSeq. So this is the structure of AbSeq. It has a poly A tail. What else is there? It has antibody specific barcode, it has a poly and UMIs and the NGS handle to do the sequencing, because our readout will be in the form of sequencing. The same kind of data you get it through the flow cytometry, that this is this much is the CD4, CD8 and all. So that is there with the help of this AbSeq technology you can get it. And there is the best part is there is no limitation because there is no other thing involved like any compensation and all. It is all oligo based technology, so you can go up to 50 or 100, many more. So now this is, because I said our technology is magnetic bead based also. So this is on the right hand side, you can see the bead where some magnetic bead, there are short DNA fragment immobilized on the bead which can capture RNA from each cell. And this is an enlarged structure of one of the fragment. It has oligo-DT and some molecular indexes, cell label and some UMIs. So this will take care of which RNA belong to which cell, and the UMI will take care of the PCR biasness. And the same way, because this era is like multiplexing, barcoding. So for what we do, we can barcode our sample before processing. So if you have 10 samples, you can barcode them, and then you can pool it, and then you can have stain with the antibody and then process it. So that way it will reduce the cost of sequencing, reduce the batch effect and all. So this, and the sample multiplexing is also having poly-A tail. Now, you can see, as I said, our system is a microwell based partitioning system. This is the cartridge, and that is the scanner, and how it will work. On the right hand side, you can see the picture of the microwells, and these green dots are the cells, and this is a picture of the scanner. And how it will work, first we load the cells. So you can see the green dots, these are the cells. With the help of automated pipette which has a fixed flow rate, and it will ensure that in one well only one cell will go. So this is a liquid engineering technology. So in that way, we first load the cells. And the limitation of loading the cell is from 150 cells to 40,000. So maximum 40,000 to 50,000. And the lower limit is if you have working on any rare disease, the only 150 cells can be processed on that. So then we wash off the excess thing, and then we load the bead. So this is the magnetic bead. This will also go and sit on the cell. Now what we do, the right hand side is the picture of the scanner. It will show me all the cells, all the wells were captured with the bead. And then what we do, we lyse the cells. So this is an enlarged picture of the cell sitting nicely, and then there is a magnetic bead. Once we lyse, all the RNA will come out and it will mount to the bead. So you can see this way. Okay. Now what we do, we retrieve all the beads, then we collect it into the append-off, and then we synthesize cDNA, and then we synthesize multiple libraries. As I'm saying that, we can characterize VDJ region, full VDJ region. So you can have your PBMC enriched with either T cell or B cell, and then you can process it on this thing. And then further on, you can see how many cells I have loaded, and then with the scanner, it will tell me the multiplet, viability and all. At this step only, you can decide whether to go for the sequencing or not. So because the multiplet rate should be very low, less than 10% if we are talking about the single cell. So this is the advantage of having the scanner, that give me the visual workflow, help me in troubleshooting and all. And the advantage is also that this technology advantages that it is having the superior cell capture rate, low multiplet rate, and we can go beyond PBMCs. You can use tissue sample, you can use cell lines, multiple things together also. So what does it mean by the capture rate? Means if I'm loading 10,000 cells, how many cells are going to be captured? So that has been proved by various R&D, and you can see in our brochure also that our capture rate is very good. It is more than 80%, and the multiplet rate is very good. It's less than 10%, even if you are loading more than 40,000 cells. So our maximum limit is 40,000. And as I said, this is the data of all the published paper, where the people have used different kind of cells, like from CAR T cells to CD4 cells and different kind of tissues for our technology, and their capture rate is also mentioned here. And we can use different kind of cells like neutrophils, Jadkat, different multiple kind of cells together also. So now, in the context of this workshop and this symposium, like to characterize the immune repertoire. So generally, when I was doing PhD and when I was in AIMS, how we produce a monoclonal antibody is like we do the single cell sorting using FACS with the antigen, and then we sort in single cell, and then we isolate the RNA, and then we made the antibody. That way is the way. But with this technology, where you can have a pool multiple, like there is how many cells you can sort at a time is 96 cells at a time. And in PBMC, as you know, because of there are many things like antibody secreting cells are very less. So there is less chance of having those cells also. So in case if you are using this technology, you can go up to 20 or 30,000 cells, enriched PBMCs, and then you can further characterize it. So there are two approaches. Either you do the T cell receptor profiling, or you can do the B cell receptor profiling along with their immune profiling, like, and the transcriptome also. Like what could be, when this was my clonotype, in that clonotype, what were the genes, downstream genes are expressed at that time. So that data you will get it. So this is the single workflow or BCR design. You can see this kind of a data you will get it, where it will tell me the full length, all the CDR regions, as well as if in this, experiment they have used immune discovery panel, which is a cocktail of 30 antibody and some 15 extra antibodies also. So you can characterize your naive cell, your memory B cell with the transcriptome, as well as what is their clonotype, what is the heavy chain, light chain, what was their sequence, and in further back you can further identify what could be the epitope of that. So that is kind of antibody discovery. Now if you've made the antibody, now how to characterize, you wanted to see the immune response. So that way also you can treat your subject of either mice or human or something, some data, and then you can see the immune response through this technology also, like what could be my protein profiling, how my immune system is regulating all those. So whether, if I'm expecting that this immune population should be going up, or then, so what that kind of application is also can be done with this technology. So same way, if you wanted to check the TCR identification, that also can be done. So that's all. This is one of the paper which is published in the year 2020 on the COVID, where they have checked the different kind of a population based on. So the advantage of doing this multi-omics technology is that we are doing the unsupervised clustering. We are seeing what could be the different population, not only based on one dimension. That is why I said it's a high dimension, not only based on the protein data. Here we are integrating both our protein and gene data. So that is a different kind of a dimension. So going deep down and then checking. And then we can have multiple things, like we can check the spheroid, different kind of population also. So this is the, and we have a stall here where we kept the machine of the Rhapsody Express. So if you have more question and all, you can come to our stall, you can see the machine, and we can have a further discussion. So these are the takeaways for the novel discovery, to characterize the immune system, and to characterize your full length TCR, BCR clonotype. Our technology, you can use it. Thank you. I'm happy to take any question, or if you want, you can discuss later also.
So thank you, Dr. Romsha, for such a wonderful lecture. I think this technology will be very useful, because when we do a single cell sorting, we find that out of these 96 cells, only 20 or 24 pairs we are able to amplify, but remaining 60 we are not able to amplify. Yes. I think maybe those are the important clones that we are missing. I think this technology will be really helpful for that.
And many times it depends upon how much binding, how much fluorescence it is giving with the flow cytometer. So if it is giving a good binding, then only we are going to sort it. If it is a low binding, we clear it off. So, but here we are having multiple cells together, and you can identify the clones.
So I will encourage the students to just maybe go to the counter and just have a look on the machine, because this is a very useful instrument. I personally feel that, because single cell sorting is coming in next generation omics for the both proteomics, transcriptome, anything. So please.
Hi. I'm Jeevita, product manager for Stem Cell Technologies.
Yes.
And I have one simple question. So you said about magnetic bead. So will that hinder the cells?
No. No way, because this magnetic bead is having oligo-DT. Whatever we are making the libraries and all, that first we have RNA, this poly-DT RNA. What we are making the cDNA. We are not using the magnetic bead further. We are taking out of all the cDNA. Once we made the cDNA, we denature it, we take out that strand, and then we made the library. So there is no hindrance of this magnetic bead.
So even after washing, there will not be any magnetic particles?
No problem. Yes.
Thanks.
Thank you.
Thank you, Dr. Doshma. Our next speaker is Dr. Subair Vishwas from GeneScript. He will be talking about integrated drug discovery.
Dr. Subir Bishwas did his PhD from CDRI in Life Science. At GeneScript, he is responsible for developing and building strategic alliances for CDMO business in biotherapeutics and gene therapy across India. We welcome you, sir.
My presentation. फुल स्क्रीन कर सकते हैं ना? It's not coming as full screen. So good afternoon everyone, and thanks Dr. Rajesh and Dr. Shri Kumar for inviting us and giving us an opportunity to talk about our portfolio of accelerating drug discovery through antibody engineering. And platform has already been set by Dr. Tripti and Dr. Nair. So let's see how we can go ahead and look into the trending approach of biologic discovery and the antibody engineering. So as we all know that the biologics or the therapeutic antibodies fall in two categories, innovator molecules as well as biobetters. So this is a graph which is showing the discovery or the life cycle of a biologics. So if you see almost from this pipeline where it starts at the preclinical stage up to the NDA filing or the lead, almost the survival rate is very less. Almost 5% of the antibodies what we generate during our research and development stage, but ultimately it reach only the 5% of those repertoires finally lead into a drug. And the reason for that is the antibodies what we are developing, they should have the intended purpose of giving the therapeutic or the mechanism of action has to be followed. So during the antibody development, what we generally follow is the functionality and the developability of the antibody. So when we talk about the functionality, we look for the affinity, we look for the in vitro potency as well as the cross reactivity, because these are the, if this falls into the required, what is the required for the mechanism of action of that antibody, then we can think that whatever leads we have generated during our research and development, those leads can be converted into some final drug targets. And during the developability, we check for the chemical stability, solution property, and the physical stability. So we will see in detail now that the antibody structure, what we are looking since morning, the Y-shaped structure having the Fab as well as FC, how the amino acid sequences in those Fab or FC region can, if we engineer those as per our desired property, how they can lead into a better drug candidates. So in antibody engineering, either we can do the engineering for a Fab region or we can do the engineering for the FC region. So in the next slide, we will now see what are the approaches of doing either this Fab engineering or the FC engineering. So this is a common workflow which everybody knows, that during the discovery we find out all the hits as an antibody. Like we have seen many presentations here that we will develop the antibody, but we don't know that which antibody is going to be our final candidate during the commercialization. So next, so what we do next is we first check those antibodies what we have developed for the functional assays. Like we have seen talk from Dr. Sorry, I forgot his name. Sorry. Dr. Sehgal, that how he has developed that antibody and did all the functional testing. So that is a second workflow stage where we do the functional testing. And after getting those functional results, we go for the antibody engineering, which is the third stage where we want that by mutating the residues or by grafting those Fab regions, we get our desired antibody. And the last but not the least is in vivo efficacy, because that is a must. If the antibodies get into their desired efficacy and safety potency, then we have some confidence that whatever we are developing can go for commercialization. So in the approach, the first one is antibody humanization. So why we do humanization is, let's say during the discovery we have discovered we have generated the antibody in mice. So all the sequences mostly will be of mice in that. So in order to make that mice variable region engineered for chimera or for the humanized, we will do the grafting of the CDR region. And let's say if the antibody we have designed is fully human, like adalimumab is the example, so we need not to do any kind of engineering there. But in case if the antibody is generated in mice, then definitely we need to do the grafting, or we need to do the humanization for generating either chimeric antibodies or the humanized antibodies. So how we do this humanization is, here is a strategy, as I am mentioning, either we can do the CDR grafting, or we can do the selectively the back mutation of the residues, so that the antibodies having those sequences, let's say alanine is there, it is not desired, we can mutate those alanine into whatever the residues is required, we can do the back mutation as well. And it's not that we will do this grafting or mutation and we will not evaluate what is the downstream effect of those mutations or the grafting is going to happen. So we need to do the immunogenicity assessment as well as de-immunization if required. So for that, it's not that always we will go for the experimentation part. We can do some in silico methods, like we have some softwares. So using those software, we can do the in silico analysis, and then we can see that, let's say we have generated some peptides, and these peptides can be putative immunogenic, or they can lead to some aggregates or solubility issues. Then after getting those kind of report, we can look into the selective removal of those peptides or the mutation of that particular amino acid residues. So this is a case study which is shown here, that let's say if I am doing some antibody engineering in that table. So parental antibody means it is a wild type which was before antibody engineering, and let's say I have done the humanization of that antibody, then I will go for the affinity measurement of that. So if you see the results which is shown through the well-known BIA-core as well as ELISA and FACS technology, if you see the comparison between parental and humanized, all the affinity values, ELISA EC50 values as well as the FACS values are correlating with each other. So after getting this kind of data, we can be assured that the parental antibody is having the same information what we have got after the humanization of parental antibody into humanized one. The second approach is antibody affinity maturation. So here what we do is we take this approach using PML. PML is nothing but precise mutagenesis library. Here we do the site-directed mutagenesis for the desired amino acids which we don't want to be in our sequence. So we can use this kind of approach to mutate the libraries, to generate the library, and then we do the screening. Again we do the affinity screening, and we can see that which sequences after that PML mutagenesis is matching with the wild type. So again you can see the affinity ranking using the instruments which are in the market already. So we can see the KD values, so whether they are falling in the wild type range or they are better than the wild type range. So this is one of the second approach for the antibody engineering, which is affinity maturation. The third one is FC engineering. So always it's not the fab game. FC is also important part of the antibody, and FC is important for the stability of antibody. So we need to take care of that FC region as well. And as we all know that the glycosylation of FC is responsible for the CDC as well as the ADCC activity of the antibody, which are the one of the most important activities of all the therapeutic antibodies floating in the market right now. So ADCC or CDC, as I mentioned, is a desired activity, and because most of the biopharma companies in India are biosimilars, so the ADCC and CDC activity has to match with the innovator molecules activity as well. In that case, let's say if I'm making a copy of my molecule, the ADCC and CDC is falling off the rate, or they are more than the innovators ADCC activity, then we have to match those, we have to make our drug which we are designing to fall into those range of innovator molecules. So for that, either we can do the FC engineering through glycan or through the again the site-directed mutagenesis. And also we know, apart from stability, the FC is also important for the clearance, the PK part of the antibody, that is blood from the circulation. The antibody has to get cleared. It's not that we got the dose and it will circulate in the body for lifelong. So the clearance has to go means that drug has to get cleared from the body, and that is also taken care by FC. So we have to look for the FC's half-life, that is T half, also we have to measure, and to match that T half with the innovator molecule, we can do this kind of mutagenesis. So this is again a case study where I am showing that the ADCC activity and CDC activity compared to the wild type versus the DE mutation or the alanine mutation, what is the effect? So what we found after doing this kind of mutation is it is in next slide. So after doing all those mutation and affinity, what we found that the variant versus positive control and the wild type, what are the KD differences? So this gives as an idea that which variant is the best after doing the mutations and all that. And last but not the least is the PTMs, that the post-translational modification like glycosylation, deamidations, all the known factors which protein gets as an added factor after the translation. So that PTMs are also one of the important thing. So because the amino acids can lead to the isomerization. So if the isomerization is happening, and let's say if that isomerization has happened in the Fab region, then probably the binding of the antigen with that antibody may get impaired. So we need to take care of the isomerization, deamidation, because deamidation leads to the basic charge increase. So the most of the antibodies, what we will be doing the scale up during the bioreactor stage and all that. Let's say if the deamidation is happening, then the basic charge impurities will get more compared to the main or the proper form. So we have to take care of deamidation as well as the glycosylation, which is important for the ADCC or the ADCC activity as well. So for that, what we can do is, first we can do again using the software, we can do the PTM analysis. Then if required, we get a risk evaluation that in this particular sequence, we get some free cysteine isomerization, or there is a probability of happening a deamidation or any kind of PTM. Then based on the risk score, we can go ahead and we can decide that whether this PTM is required. Means if it has a low risk, then you can leave that PTM, or if it is showing a high risk, then it's better to go for a PTM removal. And once you have removed that, then again the same thing, go for the affinity calculation, either using any of the kinetic method or ELISA and flow cytometer. So you get the information that after this PTM removals, these are my EC50 values which are matching with my wild type, like this. Then, as I mentioned, the amino acid sequences are also responsible for the aggregation, and we don't need aggregation in our drug product as well, which we are going to market, because aggregation leads to the adverse effect. So what we can do here again is, let's say this aggregation is happening because of that hydrophobic patches or the exposed surface. So we can again either completely remove that hydrophobic patches, or we can just go for the site-directed mutagenesis to reduce that hydrophobic interactions.
Dr. Subair, can you please?
Sorry? Yeah, this is just 2 slides. So again, after doing the hydrophobic mutation removal of those amino acids, we can calculate the KDs and see if those are falling in the range of wild type. And this is my last slide to summarize that what we can offer you from GenScript ProBio. So we provide all the 3 services what I have mentioned here: antibody humanization, maturation, as well as the PTM removals and all that. So thank you, and I'm open to have any questions, if any. Otherwise we can discuss during lunch.
Thank you, Dr. Subair. We are concluding our technical section.
Yeah. So we are coming to the finish of our first half session. So we are requesting Dr. Anismrita to hand over a token of appreciation to Dr. Romsha Kumar. We are requesting Dr. Ashwati Raj to hand over our token of appreciation to Dr. Subir Bishwas. We are requesting Tripti ma'am to hand over a token of appreciation to Dr. Anusmrita Lohan for chairing out the session. We are requesting Devendra sir to hand over a token of appreciation to Dr. Ashwati Raj for chairing the session. So I know we are all impatiently waiting for the lunch, but can I have your 2 more minutes? Like, we would like to thank our industrial partners who have put the stalls outside. So we would like to thank Ipendorf, BD, Biorad, Cellgenics, Beckman Coulter, Crescent, Aravind Herbal Labs, Thermo Fisher Scientific, 50 Plus Scientific, Sartorius, Biotech India, Biolinks India, Art Lab, Promega, Biogene, Merck, Zelly Biotech, and Trivandrum Surgicals. Our prize money is sponsored by Canara Bank. So we are urging you all to visit the stalls that have been put outside. It will be informative, as well as you can get more ideas when you go back to your labs or colleges for your future work. So we are breaking for lunch now. We'll have 30 minutes of quick lunch, as we say, and we'll come here back by 2:15. So thank you.
हमें एक नई एजेंसी स्ट्रक्चर करनी है फॉर रिसर्च एंड एनालिसिस ऑन इंटेलिजेंस अफेयर्स एंड नेशनल सिक्योरिटी, और मुझे पता है कि अगर उसमें तुम्हें शामिल नहीं किया गया देन इंडिया विल बी एट अ लॉस। मैम, वो मूर्ति और मेरे बीच एक टग ऑफ वार चलता रहता है, cabin ops versus grassroots intel. So I think that's the reason he's hesitating.
Raw will be best at both. India के best spies.
World के best spies, ma'am.
We are
worst piece of advice you could give to a first generation entrepreneur or to a family business owner is that you got to try it all by yourself. You got to figure it out.
Hello, 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 check, check.
എല്ലാവരും കണക്റ്റഡ് ആണോ ഇമെയിലാണെങ്കിലും കേൾക്കാൻ പറ്റുന്നുണ്ടോ? Good afternoon all. I hope you all had a wonderful lunch and we are expecting some freshness even in the afternoon session. So we are moving to this session 3 where we'll have Dr. T.R. Santosh Kumar sir and Dr. Binod sir for chairing the session. We are welcoming you both to the chairs. Dr. T.R. Santosh Kumar is working as Scientist G in Rajiv Gandhi Center for Biotechnology. He received his PhD in Tissue Engineering from Sri Sitra Tirunal Institute of Medical Science and Technology in 2000. He is leading his team to understand molecular mechanisms of chemoresistance and tumor recurrence in triple-negative breast cancer. He has published several papers on live cell-based assays for identifying molecules that specifically target key pathways in cancer such as cell cycle, angiogenesis, HIF, and tumor stem cells. Dr. Binod Kumar received his PhD in microbiology, particularly virology, from the Vallabhbhai Patel Chest Institute, University of Delhi in 2014. He then joined as a postdoctoral fellow in the Department of Microbiology and Immunology at Rosalind Franklin University of Medicine and Science, USA, where he has subsequently promoted to the Senior Research Associate position. Dr. Kumar has demonstrated expertise in the area of entry and trafficking induced host cellular signaling and associated innate immune responses against herpes virus, hepatitis, and human coronavirus. His current research institute include mechanistic studies on viral host interactions. His laboratory conducts research on innate immune pathways that get activated in response to viral infection or vaccination using VLPs and develop potential antiviral drugs against emerging viral infections. So welcoming both of you for the chairs, and we are handing over to you to introduce the next speaker and the topic.
Good afternoon all, and thank you for a nice introduction. And now we are starting the post-lunch section. And our first speaker is from this institute, Dr. Mohanan Valliyaveetil. He's a senior scientist, and also he's going to talk to us on a very important topic.
Sir, may I help you in taking that?
Huh? Okay, no problem. No, just basically only title actually. Okay. At least, give that honor to me. Okay. Anyway, you go ahead with the introduction. But anyway, he is going to talk to us on techniques for isolation of monoclonal antibodies using phage display, hybridoma, single memory B cell technology, their advantages, disadvantages. It's going to be an exciting talk. Over to you, please.
Dr. Mohanan Valliyaveetil is working as a senior principal scientist at Institute of Advanced Virology, Trivandrum. He earned his PhD in biotechnology from Cochin University of Science and Technology. He completed his postdoctoral studies in virology at University of Kansas Medical Center and at Chicago Medical School, Rosalind Franklin University of Medicine, USA. His current research interests focus on the interaction of viral proteins with host cellular molecules and the development of antibodies which functionally block the interaction during virus infection. We are officially welcoming you to the lecture series, sir.
Good afternoon, everyone.
So, first of all, I would like to thank the organizers, Dr. Rajesh and director for inviting me, though I am a part of the IAV family. So, the title is Monoclonal Antibodies in Therapeutic in a Special Context to Viral Infections. So, you may find some deviation from the title because from the previous speakers you have already heard about, learned about the antibodies utilized by viruses and how they interact with the antigens, antigen-antibody interaction and the epitope formation, and other things have been already been learned from the previous speakers. So, so let me start with some introduction about viral diseases. There are hundreds of viral diseases caused by viruses, and these diseases include some minor infections or mild infections which include rhinovirus, common cold, or common warts are examples for minor infections which are barely noticeable. And there are some infections which are called moderate infections. These infections are caused by moderate viruses which cause moderate infection, which include dengue virus and chikungunya virus. In addition to this, there are some viruses which cause serious life-threatening diseases. These viruses include Ebola virus and Nipah virus, and these viruses cause high mortality rate, and community spread is also very high compared to the moderate infection, where the mortality rate is very less. Serious life-threatening infections causes very high mortality rate. In addition to these three types of viruses, there is one more class of virus, that is oncogenic virus. Oncogenic viruses normally cause different kinds of cancers. HPV and EBV HCV are the different kinds of oncogenic viruses, and these viruses cause almost 18 to 20% of cancers worldwide. So that's a brief introduction about the viral diseases caused by diseases caused by viruses in humans. Now, if you look at the pathogenesis of virus, it includes several mechanisms. The first one, step is viral transmission into host. That is, virus is transmitted from one person to another through several portals of entry. These portals of entry include respiratory, alimentary, urinary tract, anus, and skin. And this is followed by replication at the entry sites. Once the virus is implanted on the cells, the virus attaches to the cells and enters into the cells. And inside the cells, genome replication, assembly, and new viral particle formation occurs. And that is followed by the spread through the body. Spread through the body means once local information, replication happens, it infects its neighboring cells, and from there it is released into the lymph nodes and blood vessels. And that is followed by the spread of virus throughout the body. And when the virus spreads throughout the body, it can cause diseases by targeting its target organs. And this is caused by several mechanisms which include cytopathic effect, changes in host cellular processes, autoimmunity, immune suppression, and persistent infection. Finally, the viral particles are released into the shedding site. That is, once they complete their life cycle, these viruses escape from the cells by cell lysis or by budding through the plasma membrane. In both processes, the viral particles are released into the external environment, and from the external environment, these viral particles infect another host, and this life cycle continues. That is how viral life cycle pathogenesis occurs. Now, I'm moving on to the receptors on the cell surface. I said that once the virus is implanted on the cell surface, it attaches to some kinds of molecules. So what you can see here is a cell membrane which contains several different kinds of molecules. These molecules include proteins, lipids, and carbohydrate molecules. And one of these molecules is utilized by virus. So a specific virus utilizes a specific receptor. If that specific receptor is absent on the cell membrane, then the virus cannot enter inside the cells. Those cells are called non-susceptible cells. But the viral, if the cell contains receptors, then it can attach to receptors and then the virus can go inside. Those cells are called susceptible cells. And this presence of receptors also plays an important role in tropism. For example, HCV target liver cell. HCV doesn't affect other types of cells because its receptors are present in the liver. Similarly, other kinds of viruses also, you can see that certain viruses attack the respiratory system, certain viruses attack gastrointestinal system, certain viruses attack brain system. This is due to the fact that the presence of certain receptors. If these receptors are not present on other organs, then they will not affect the organs. So now we saw that viruses attack a particular receptor on the cell surface, and these receptors are divided into two types, that is attachment factors and specific receptors. Attachment factors are used for initial contact with the cell surface. They are a platform for the virus to attach to the cell surface. And they also help to concentrate the viruses on cells. And this binding is non-specific and it is based on charge. It is based on electric charge, which means that positively charged viral particles interact with the negatively charged cell surface molecules. So this is not a very high affinity interaction. It is a very low affinity interaction and it is mostly charge-based interaction. And in addition to attachment factors, there are two kinds of receptors. These receptors are called specific receptors. Specific receptors includes primary receptors as well as secondary receptors. So now specific receptors are high affinity receptors, and they can attachment of specific receptors to the viral particles induce conformational changes in the viral particles. These conformational changes activate signaling pathways as well as certain other intramolecular arrangement that leads to the entry of the virus. Virus. So now we saw that attachment factors is followed by the binding of virus to the primary receptor, and once the virus is bound to the primary receptor, certain viruses use a second receptor which is called a secondary receptor or co-receptor. Secondary receptors are to make sure that the virus is bound to correct cell. After binding to the secondary receptor, fusion with the cell membrane occurs and the viral particles genome is released into the cytoplasm of the cell. So this is how viral infection occurs, and by utilizing different types of receptors. So now once the viral particle attaches to the cell surface receptors, it can enter the cells by two methods. That one is by fusion and the other is by endocytosis. By fusion, the cell surface receptor molecule attaches to the virus, and after attaching to the virus, conformational changes takes place, and this conformational changes leads to fusion of the viral particles with the cell membrane, and the viral capsid is released into the cytoplasm. Whereas in the endocytosis, the viral particle is endocytosed by utilizing the signal pathway induced by the cells, and the viral particles then in the endosome fuses with the endosomal membrane, and from the endosomal membrane the capsid is released into the cytoplasm. And there are different kinds of endocytic pathways. These endocytic pathways are divided into two types, that is phagocytosis and macrophytosis. In phagocytosis, cell deformations engulf large viral particles, whereas in macropinocytosis, they to target a viral proteins. So I have selected SARS coronavirus RBD domain as the test protein. So in this you can see that the virus consists of SARS coronavirus consists of 4 structural proteins, which include spike protein, then membrane protein, nucleocapsid, and envelope protein. And the virus initially binds to the cellular receptor ACE2 with the S1 protein present on the membrane of the virus. This S1 protein has several subunits. The RBD, or the receptor binding domain of S1 protein, binds to the ACE2 receptor, and this receptor binding induces several cellular conformations, and that is followed by the cell signaling pathways, and virus is endocytosed. Endocytosed viral particles releases the genome, and this genome then multiplies and forms new viral particles. This is briefly about the life cycle of SARS coronavirus. So in RBD is the portion which really binds to the receptor of the virus. If you look at the spike protein of a linear sequence of the spike protein of coronavirus, you can see that it has an N-terminal, a C-terminal, RBD domain S1 and S2 domain. RBD has 211 amino acids. To create antigenic sites on RBD domain, we can use an antigenic plot software. So when we use an antigenic plot software, we found that 7 peaks, this yellow represents the antigenic sites with a high probability. All these yellow peaks represent antigenic sites with a high probability. In addition to that, we also found 5 peptides with more than 12 amino acids. So these 5 peptides with more than 12 amino acids can be used for producing peptides. And the peptides produced can be injected into mice, and from this mice, splenocytes can be isolated. And from the splenocytes, B cells which produce antibodies are isolated. These B cells can be fused with myelosoma cells, and that produces hybridoma. And from the hybridoma screening, the cells that produce monoclonal antibodies are again screened, and they are expanded and produce in huge amounts. And these monoclonal antibodies are further purified by protein A or G purification methods. Now, how do we do neutralization? How do we know that this antibody has the ability to neutralize virus? So in this case, mix the virus with the antibody. You can see that in one case, the virus is not mixed with virus. In the other case, virus particles are mixed with antibody. The virus particles that are mixed with virus antibody cannot go inside the cells, because all its glycoproteins are occupied by antibodies. Whereas in the case of the untreated virus, the viral particles goes inside and infects the cell. So this is the method, this is how the viral particles neutralization assay is done. So that's all for today, and thank you for your patient listening, and thank you. And if you have any questions, please.
Thank you, Mohanan, for giving an overview of antibody generation and viral pathogenesis. Since we have time, we can have a couple of questions from audience. Please. That is also. Yeah, if there is no specific question from that, I have one question actually. I thought you are explaining the viral internalization process by macroautophagy, pinocytosis, macrocytosis, then collagen-mediated and independent mechanisms. My question is that these are the physiological internalization mechanism existing in all mammalian cells. And is this, all these pathways are there in any cell actually. So if for a particular virus, this is triggered only one pathway, or it coexists all things, everything. For example, SARS-CoV-2 infect, internalization is mediated by a receptor mediated, clathrin mediated. All this will you encounter, or it is virus specific? What we know about?
That depends on the type of virus, because in certain cases, as I mentioned, direct fusion occurs. In direct fusion, the viral particles fuses with the cell membrane receptors and enters inside the cell. So that is called a pH independent fusion. But in certain cases, the viral particle requires pH for conformational changes, because the viral glycoprotein undergoes conformational changes. So that is done by some pH change. In that case, the virus particles endocytose. Inside the endosome, the pH will be lower than the cytosol. So at this lower pH, the viral particles, glycoprotein undergoes conformational changes and then fuses with the endosomal membrane and releases viral particles. Whereas in the case of macropinocytosis...
No. My question is specifically I wanted to know, is all coexisting actually? If when a pathogenesis happens, whether the virus will utilize all these machineries of internalizations of the cells, or some virus only will go through receptor, rest of all the pathways are not being touched. That kind of knowledge is there or not? Because if so many mechanisms are there, means obviously targeting of one mechanism, even sometimes neutralization may not be helpful, because there are other mechanism which is independent of receptor mediated will go on actually. That is the reason why I asked, because at least for SARS-CoV-2 we know that neutralizing antibodies are were capable of completely inhibiting at least in experimental model as well as certain clinical situations the infection.
Okay, so in certain viruses, if the virus goes by clathrin-mediated endocytosis, if you block clathrin-mediated endocytosis, it can go by macropinocytosis. But the virus size is also important, because in macropinocytosis...
Okay. So virus biology also affected.
Yeah, some viruses cannot go by caveolin-mediated endocytosis, because viral particles less than 50 nanometer can go by caveolin-mediated endocytosis.
Okay, thank you. So we will have more questions during the tea time, and over to you. We are going ahead with the next one, isn't it?
So next speaker is Dr. Rajesh. He is going to talk about the techniques of isolation of monoclonal antibodies using various ways, and show their advantage, disadvantage, and the future prospects. Over to you, Rajesh.
Dr. Rajesh Kumar received his PhD degree in 2014 from All India Institute of Medical Sciences, New Delhi. He was inducted into THSTI in 2014 and has been worked immensely on various aspects of translational virology and has contributed significantly for the bench-to-bedside solutions in the field. He has successfully isolated human monoclonal antibodies against SARS-CoV-2, which implies the application of generated monoclonal antibodies as available reagents for accessing the antigenicity of vaccine candidates. He is presently working as Principal Scientist E2 in Institute of Advanced Virology. His research interests focus on development of antibody-based therapeutic interventions for emerging and re-emerging viral disease. I welcome you, sir.
So good afternoon, everyone. So actually I prepared my talk based on 30 minutes, but I think Dr. Sehgal, Dr. Nair, Tripti and Dr. Mohan has already introduced most of my slides, so I will try to finish in 10 minutes time. Okay. So as we know, for any viral intervention, any disease, there are three major interventions. First one is that either we can make the vaccines, as the best example is the SARS-CoV-2. The other thing is that we can make small molecular drugs. And the third that has recently emerged in the last 10 years is the monoclonal antibody-based therapy, especially in the case of viral infection that has emerged lastly in the last 5 years. So I will skip some of the slides. So I directly come to what are the different methods of monoclonal antibody isolation, what is their advantage, what is their disadvantage, in which case we have to use which method, and what are their applications. So based on our necessity, we can choose our method that can be used for the isolation of monoclonal antibody. So there are 3 different methods for the monoclonal antibody. The first one and the traditional one is the hybridoma technology. And the second one is the recombinant technology that mainly include the far display technology. And the third and the most recent one is the single memory B cell technology. So first I will talk about the hybridoma technology. This is the most traditional technology that was established by Professor Kohler and the Milestone, and for which they have been awarded the Nobel Prize. So here we can immunize the animals like a mice, rabbit or any animal. Then we can take the spleen cells and fuse with the myeloma cells, and the resulting hybridoma has a dual property: continuously growing property from the myeloma cancer cells and the antibody secreting from the B cells. So these are the fusion possible partners. Here you can see there are 3 possibilities: either 2 B cells can fuse, 2 myeloma cells can fuse, or a B cell or a myeloma cell can fuse. So we put the selection marker in such a way only the B cell fused with the myeloma cells can grow in the system and other cells die. So I will give an example of this technology. Dr. Deepak has already mentioned about that, how these hybridomas are produced. So in our lab, actually we were mainly targeting to develop monoclonal antibodies against the SARS-CoV-2. So as we know, in the SARS-CoV-2, RBD is the main protein that binds to the human AC2 receptor. So if we are able to make a monoclonal antibody that has a more affinity to RBD rather than the human AC2 receptor, so we will be able to block the binding of the virus to the host cell. So keeping this thing in mind, we developed the RBD protein in the mammalian expression system. We immunize the mice, and then we give 3 times boosting of the mice, and then we did the simple ELISA experiment, and we found that all the animals were able to give a good high titer of binding antibody. Here you can see in the first graph. And the second one was we have 4 mice in each group. The next one was which is our best mice that we can select to make the monoclonal antibody. We collected the sera from the mice, and then we did the neutralization assay against all the variant of the concern. And we found that there are 4 mice, but animal 3 was giving the best neutralizing antibody response either. So mainly it was on the polyclonal level, but we found that this mice can be a best candidate for making these mice monoclonal antibodies. So we took this mice, and then we fused with the myeloma cells, and we developed a large panel of monoclonal antibodies. And among these antibodies, we selected 2 antibodies that were able to neutralize all the variants of concern, including the Omicron variant. So then we compared the neutralization efficiency of this antibody with the already approved, FDA approved monoclonal antibodies, and we found that the monoclonal antibody that were developed in this study was far better than the FDA approved monoclonal antibody. So after that, we tested the cross-reactive binding of this monoclonal antibody with a different RBD protein of all the Omicron, all the SARS-CoV-2 variant starting from alpha to Omicron, and we found that the antibody were binding with all the variants. There was a little bit slight, you can see, variation in the binding, but antibody was able to bind to all the variants of the concern. So then we infected the viro cells with the live viruses, and then we took the P4A2 antibody and we did a simple immunofluorescence study, and here you can see we can count these foci in the form of dots, and lower are the value. We can see the P4A2 antibody was able to recognize all the variant of concern infected cells. So here we proved that this is a broadly cross-reactive antibody and a broadly cross-neutralizing antibody. So what happens when we make a monoclonal antibody? So this, all the testing was done in the in vitro lab. Either it was with the pseudovirus or the live virus. But what happens in the real case scenario? this antibody can be used for the therapeutic purpose, the first step was to do the animal study. So we took the AC2 expressing mice, that is AG129 mice. So first we give the antibody, then after 12 hours or 24 hours we have given the virus. And then here you can see we found that all the mice there was no death in any of the mice. We found that this antibody was very protective when it was given before the virus challenge. But what happens in the real case scenario, patient comes to the hospital after the virus infection. So then we tested as a therapeutic antibody. Suppose a patient is infected with a SARS-CoV-2, same here we took the mice. First we infected with the virus and then we added the antibody after the 12 hours, and we found that in the same case all the mice were protected, and we found that this antibody was very effective both prophylactic and as a therapeutic agent. So then comes, if all things are good, then means what are the limitations of this? Actually what happens, this was a mouse monoclonal antibody. We cannot directly give to a patient because this will recognize as a foreign antigen in the human body. So the main limitation was that we have to humanize that antibody. If we could have generated this antibody from a human patient or a human source, so we can directly use this antibody after some modification for the patient use. So that was the major limitation that we found of this study. But if these antibodies we make against other antigens, they are very good antibodies that can be potentially used for diagnostics and the other things. So here you can see, if we give a mice antibody to a human, so the half-life is only 1.5 days. So means after every 1 day we have to infuse the patient with the monoclonal antibody, and the chances of immunogenicity are very high. The second thing is that we can make a chimeric antibody. We can take the variable region from the mouse and the other regions will be from the human. The half-life increases to the 10 days. But again the problem is that immunogenicity will be there because 25% of the content is from the mice. And the third one is the humanized antibody, where the complementary determining region mostly will be from the mouse and all the grafted sequences will be from the human. So means almost 95% of this antibody is from the human nature. So the half-life is 21 days. The last option and the best option is that we can directly make an antibody from the human samples. So next question arises, why we are not making antibody directly from the human by using the hybridoma technology. So what happens? So during my PhD, I was working on the HIV viruses. So we thought we will make a monoclonal antibody directly from the infected patient samples. So we almost selected 33 HIV-1 infected patients, and we took 320 million cells from those HIV infected patients. We did a fusion experiment, and it was unbelievable that after doing 4 years of getting this fusion and everything, we got 3 monoclonal antibodies, and all 3 were non-neutralizing antibodies. So after the 4 years of hard work, we landed with nothing. Then one of the major challenges is that for mouse, we have a fusion partner. We have a mouse cancer cell that can fuse well, and they fuse and there is a chromosome stability. But for human, there is no such good fusion partner. So what happens when a mouse cell fuses with a human cell, there is a chromosomal instability. So that is the main reason. And there are also some hybrid partners, but they are also not very successful. And other is that there are some retroviruses in the mouse myeloma cells. If we fuse with the human cell, all those retroviruses will also come to the human B cells. So there are some challenges, and other problem is that for HIV it was okay because it's a chronic disease, but for other disease it's not possible we can immunize a human. There are a lot of ethical reasons that we cannot immunize a human. So all these limitations are with the hybridoma technology, especially for the development of the human hybridoma. So then what happened? Parallelly, we started working on a technology that known as the phage display technology. So one of the major advantage of this technology is that it does not require animal cell culture. It does not require any sophisticated instrument. Here you just need bacteria to start your work, and the whole cycle moves around bacteria and bacteriophages. You just buy a bottle of media of 2,000-3,000 rupees and you can start your work. So that is the main advantage of this technology. All scaling part mainly happens in the bacteria. Here variable antibody genes are cloned into the plasmid vector. So they display mainly antibody in the form of single chain or Fab on the surface. So this technology was developed by Professor Winter, and they recently awarded Nobel Prize for developing this technology. So when we look at the strength of this technology, so this technology till date 25 fully human monoclonal antibodies are approved by the FDA that are fully of human nature. So out of these 7 of these are developed by the human antibody library and 18 are developed by the transgenic animals. So if you look at these 7 antibody, the first one Dr. Lalit Kumar this mentioned is taken care 50% of the global market revenue of the monoclonal antibodies. So these antibodies were the first antibody that were developed by this far display technology, and there are number of antibodies that are developed by this technology platform. If we really look at the clinical trials and phase 3 of the recent years, there are 10 antibodies that are developed by the transgenic animal, 6 are developed by again this far display technology, and 4 are developed by the single memory B cell technology platform. So this is the overview of the far display technology. So here we can make a library directly from a patient sample, or the other advantage is that we can take a healthy individual. So what happens when we have any infection? Our immune system has the capability to recognize those infections and combat those infections. So here I will. So when we construct a library from the healthy individual, so that library is mainly known as the naive library. So this library is naive. So you can screen this library against any antigen, whether it's a viral antigen, whether it's a bacterial antigen, whether it's an auto-antigen of the human body. This antibody library has the capability to recognize any kind of the antigen. So that is the major advantage of making the naive human library. So, but one of the major challenge with this antibody library is that human antibody reporter, we have to make a big reporter of 10 raised to power 9 to 10 clones. So that is a major limitation, but once we have a library, so single library is more than enough to screen antibody against a number of antigens. And second library is that suppose a person is infected with SARS-CoV-2 or HIV. So we can directly, already the immune system is mature there. IgG antibody secreting B cells are there. So we can directly take those B cells, take IgG specific primer, and we can make an immune library. So we do not need a library of 10 raised to power 9 to 10. Even 10 raised to power 6 to 7 library is good enough to get a very handful number of monoclonal antibodies. So that is the advantage of the immune library. But the main disadvantage is that every time you have to make a new library for each disease. So that is a major disadvantage of the immune far display library. So during the SARS-CoV-2 pandemic, when it started, so it was not clear whether it's a BSL-3 or something like that. So patients' samples were coming and we were handling in the BSL-3. So at that time we have a library from the Professor Winter lab who got the Nobel Prize for developing this far display technology. And we used this already existing library. We did the biopanning and we got around 8 to 10 monoclonal antibodies, and we did a 3 round of different selections. And we found one of the monoclonal antibody that was pretty well binding to the SARS-CoV-2 RBD. But one of the interesting things that Dr. Deepak also mentioned, that affinity also matters a lot. Because if your antibody has a good affinity, it can bind to the virus before it binds to the receptor. Then we found that this antibody has a nanomolar affinity, but the K-on rate was very slow. It was binding very slowly, but once it binds, it dissociates very slowly. So that was the case. When we incubated 1 hour with the virus, there was no neutralization. But when we incubated with 2 hours, it was able to neutralize the SARS-CoV-2 infection. So then we engineered this antibody in different formats, and we did a small angle X-ray study, and we found that this antibody, single chain antibody, binds with 3 epitopes on the SARS-CoV-2 spike protein. And this whole process was completed in a 2 weeks time. The pandemic started in 2 weeks time. We have the antibody, we did all the work, and in 1 month of the time the paper was online. So that is the fastest technology. Once you have a library, you can make any antibody. So that is the major advantage of this technology. You do not need any human patient sample. It avoids all the biosafety regulations. You directly take the library, you have the protein, start screening and make the antibody. So that is the major advantage of this technology. And the other advantage is that some of the human antigens, like in cancer, some of the auto-antigens and other antigens, if we immunize in the mice, there are chances that we will not get a good response. But these naive libraries can also be used for isolation of monoclonal antibody against the auto-antigens also. And the other wonderful class of antibody that has emerged in the last 2-3 years is the nanobody. So camel and shark contains a novel class of antibody. Human contains a full length antibody, but camel contains these small antibodies. They are very tiny antibodies. They can even go and bind to the cryptic virus epitopes. So these are the advantage of these antibodies. They are very small in size. They can go and penetrate everywhere because the size is only 15 kilodalton of these antibodies. But we found that these antibodies are even good, but again these antibodies are from camel or the shark. We have to humanize these antibodies. Although they are lower in size, they have the lower immunogenicity compared to the mouse antibody. So recently one of the monoclonal antibody has been approved by the FDA, that is for the blood clotting factor. So this technology is also gaining attention because if we have these antibody and we specifically target to make antibody drug conjugate. Suppose we want to conjugate antibody with a drug, and then we want that this antibody can go and bind to the viral infected cells and then release the drug there. So such kind of applications are more important in case of the shark and the camel antibody. So by this I will end my talk.
Thank you, Rajesh. It
Yes.
was a very informative talk. We can have few questions from audience.
Are there any disadvantages of fast display?
Yes, sir, there are disadvantages of the fast display technology I mentioned. If we have a name antibody library, so the antibody that we will get may be of not very high affinity. They have a good affinity, but their own rate might be slow. So these antibodies sometimes need engineering. But if you make an immune library, definitely you will get a very good class of antibodies.
Rajesh, what are the chances if you are using the immune library, which you have prepared for one specific class of disease, in order to get good antibodies for other diseases? Because the basic repertoire is same.
Actually, basic repertoire is same, but the primers that we mostly use in the immune library are IgG specific. So mostly they will catch only that are IgG specific. If you talk about the name library, we design our primer in such a way they can also take the IgM class of antibody that are the name. We can screen that. There is always a probability that we can get a chance, but chances are less.
So basically you are saying that from naive library you can get
With
IgM.
a naive library with a good diversity. That is also a second clue that we have a naive library, but diversity should be 10 raised to the power 9 minimum.
So, but in case of immune library also, there is a chances that you will get a IgG also.
That possibility is always there, but mostly is that if we have an immune library and suppose we make from the SARS-CoV-2 and we are screening that, we have a very good chances that we get a very high affinity antibody that might not be needing any engineering for doing
that. Rajesh, I have a question. See, while you are actually presenting the progress of antibody engineering approach from mouse monoclonal to chimerization, then going to humanization and human recombinant. The half-life is the one that basically is increasing. What is actually the basis of it?
Actually, sir, what happens, because mouse antibody, any antigen, mouse antibody, our immune system will recognize as a foreign antigen, sir.
So that will take some time, isn't it?
Yeah, it has already been.
But for example, you said that half-life of mouse antibody is 1 day.
1.5 days.
If it is antibody-dependent elimination, you expect it
to elimination, yes, sir.
After long time.
Yes. Immunogenicity and elimination are the both pathways are there. As soon as FC portion is in the chimeric antibody, the FC portion is from the human. So elimination process will be delay. And when we make it more humanized, immunogenicity will also decrease and the elimination process will also increase. When
or
it is
some autophagic kind of,
Yes, sir.
Is it a macroautophagy or something like that? Because antibody anyway mediated, next time when you inject, obviously, I can agree. Maybe within a short period it will be eliminated.
No, the immunogenicity, I can understand. Once you give antibody, you are saying if a second time we give, so very strong immune response will be developed.
Yes.
Yeah, it's same like any antigen.
But that value is very dramatic. When it goes to recombinant, it is showing more 10 days, more than 10 days.
Yeah, if we talk about the normal IgG half-life, it's 3 weeks, 21 days.
3 weeks.
21 days, yeah. So again, it also depends on antibody to antibody.
Okay, great.
Thank you. Yeah, thank you.
So basically, it was very exciting to hear from both. Now we have one more, isn't it?
So before we move on to the next technical session, we would like to facilitate the talkers.
Now we are going to have a presentation by Dr. Harish Srinivasan. He is going to talk to us on multiplex suspension immunoassays for proper protein quantification. Over to you.
We are welcoming Dr. Harish Srinivasan to the dais. Dr. Harish Srinivasan is working as Senior Field Application Specialist, BioRed Laboratories India Private Limited, Bangalore. He has a PhD in cancer proteomics from German Cancer Research Center, Germany. He has strong expertise in bioplex and real-time PCRs. At BioRad, he is responsible for the application support of various instruments and kits.
Am I audible? Yeah. So a very good afternoon, everyone. So let us move away from the basics of monoclonal antibodies. Let us move to a little into the applications of antibodies on, say, diagnostics or research as such. So the topic that I have today is multiplexing immunoassays for protein quantification. So this is going to be the agenda of my talk. We are going to see a little bit on immunoassays and ELISAs, and we are going to move into multiplexing assays where the advantage over ELISA is seen. Okay, so any molecule, any immune molecule, let's say antigen, antibody, or any immune molecule, which is involved in getting an assay in place to quantify immune molecule in a system or to quantify antigen in a system. So this is called as immunoassay. So an immunoassay is used to detect any analytes that are present naturally in the body in the form of hormones or ligands. In case of cancer, it is cancer antigens or any antigen, or any analyte or any antigen that is as a result of any pathogen infection or a pathogen introduction. So these are the methods that are widely used: immunohistochemistry, immunoprecipitation, radioimmunoassays, antibody-based microarrays, Western blotting, the most widely used one, ELISAs, and then we see bead-based multiplexing assays. Okay. Before we move into bead-based assays, we'll see a little bit on ELISAs, wherein you use antibodies, especially monoclonal antibodies, for the reason that monoclonal antibodies are more specific, and specificity is something that is very, very important when you want to when you go into diagnostics ideally. Okay, and ELISAs, basically enzyme-linked immunosorbent assay, are of many different types. So direct ELISA, where antigen is coated on a planar surface over which the antibody labeled with a particular binder is going to be present, and the substrate is going to be cleaved, and then you will see the absorbance, and then you'll measure the presence of analyte. So indirect ELISA is also something similar, where a secondary antibody is included to increase the specificity of your assay. The most specific thing is a sandwich ELISA, wherein you have a capture antibody and you have an antigen which is sandwiched between the capture antibody and a detection antibody. In this case, when both the antibodies are monoclonal, the more is the specificity of your assay, ideally. Okay, and whereas ELISA is mainly going to be useful in detecting antigens which are present in microgram levels, ideally. But presence of smaller molecules, or presence of these antigens or these analytes, which are mainly when a pathogen is getting introduced, like say your cytokines or chemokines, whichever results as a part of inflammation of a host. So these always occur in very low levels, picogram levels and femtogram levels in serum and plasma of the subject. So that is where the technology which I'm going to present is going to be very helpful. Okay. So we call this XMAP technology, okay, and this is developed by a company called Luminix, which is licensed by Biorad and other companies. XMAP stands for X is your unknown, okay, M is multi, A is analyte, and P is profiling. So you are going to profile unknown multiple analytes from a given set of samples, ideally, okay, and how this is going to be used. So it is very similar to ELISA, wherein your ELISA is going to be using the antibodies that are going to be coated on the plate, okay, on the surface, which is stationary, and the antigen is going to be sandwiched in between a detector antibody and a capture antibody. Okay. So what if the plate is removed and a magnetic ball is introduced? Okay. So we have a ball which is found suspended in the solution, okay, and which is going to be coupled with the antibody, a capture antibody, and the antigen is going to be sandwiched in between the detection antibody and the capture antibody, and the fluorescence is going to be detected. Okay. So now this ball, okay, this this magnetic ball which is there can also be designed in such a way that you can identify different types of these magnetic balls. Let's say that you have a basket of balls, okay. So you have green colored ones, you have red colored ones, and you have white colored ones, okay. So imagine that you are going to couple your green color balls with, say, interleukin 1 antibody. You are going to couple your white color balls with interleukin 2 antibody, and you are going to couple your red color balls with interleukin 3 antibody. So we have we can easily identify the type of the ball with which the antibody is coupled with. So same is the concept that is being used here, okay, where we have 6.5 micrometer magnetic balls, okay, microspheres, which are going to be coated with two different dyes, red and the infrared dye, okay. So as you know, any dye that is of a color, okay, has a different fluorescent signature associated with it, or the floral spectra attached with it, okay. So when we have a ball which is coated with, or a microsphere which is coated with red and infrared dye, okay, and when there is a method which can identify the amount of red dye and the infrared dye coated in different permutations and combinations over this microsphere, you can have different signatures associated with the microspheres, and this is going to be helpful in distinguishing the type of analyte which is coupled to the particular microsphere. Okay, so that is what we call as based on the bead color. Okay, we call these microspheres as beads. Okay, and based on the amount of red and infrared dye that is coated on these beads, we make different kinds of it. Okay, how many kinds of such beads are there? We have around 100 different beads associated with the two color mixing pattern. Okay, there is a little advanced mixing pattern available where a third color is also introduced. When there is a combination of three colors coated on a dye, then the flexibility increases up to 500. Okay, so imagine that you have one well. Okay, you have one well, you have one analyte, and you have one particular antibody coated in the well. So you measure one particular analyte from a single well for one particular sample in an ELISA. Okay, imagine that you have hundreds of antibodies coupled to 100 different beads which are present here. Okay, which are present here, and each and every bead is going to be coated with a particular type of antibody. Okay, so you have the capacity to detect or capacity to investigate 100 different analytes in a single well, okay, with the same amount of sample that goes into the well as in the case of your ELISA. Right. So whatever you see here is a picture from the software, okay, on the bottom. So whatever you see in the bottom, is there a pointer? So whatever you see here is a picture from the software where these 100 different bead regions are mapped, okay? And every time a well is going to be read, the beads are going to get populated in the specific regions in the map so that there is no classification error that is going to happen. So how does this technology work? The 5 basic components of this technology, okay? So you have the biological reagents. It can be the antibodies as well as a nucleic acid molecule. You have the beads, which are going to be color coded with the infrared and red dye. And you have the fluidics, which is going to carry the beads from your plate to the cuvette and to the investigation part of the instrument, okay? So which is nothing but optics. And you have the optics, which is going to be having 2 different lasers, okay? One is to detect the bead fluorescent map, okay? 2 is to detect the presence or absence of the analyte of your interest. So these are the 3 instruments that are in place from Bio-Rad. So BioPEX MagPix, which can detect up to 50 different analytes in a single well, 200 which can detect up to 100 different analytes in a single well, and 3D system which can detect up to 500 different analytes in a single well. How does the technology work? As I said, so you have 2 lasers acting in tandem. So you can see that the red color beads are lined up here. Okay. For some reason this picture is not appearing. Okay, so I'll try to explain with this slide itself. So you can see that the beads are getting lined up here, right, at this part. So when it is getting lined up in the cuvette, okay, there are 2 lasers which are focused on the beads. One is a green and the other one is a red laser, okay. So the green is going to investigate the presence of your analyte in the form of phycoerythrin fluorescence, okay. So your beads are going to be coupled with antibody X, and analyte X is going to bind to the antibody, and the direction antibody against analyte X is going to be coupled with streptavidin, okay. And there is a coupled with biotin. Your detection antibody is coupled with biotin, and there is going to be streptavidin, which is going to be conjugated with phycoerythrin, which is going to bind with biotin, okay, biotin and streptavidin combination, and fluorescence is going to be red in the form of phycoerythrin, okay. So which means that you're going to measure 2 different fluorescence here. One with the help of a green laser, which is going to excite phycoerythrin molecule and measure the fluorescence out of it. 2, the red laser, which is going to excite the infrared and the red dye that is bound on the beads, and it is going to collect the spectral signature of the beads, thereby identifying which particular bead region the analyte is bound to. Okay, so this is our detection system, where you can see the bead is in the center here, okay. Green laser is lighting up the bead as well as the red laser, and the fluorescence from red laser or the red dyes are being captured as classifier 1, classifier 2, and a double discriminator avalanche photodiodes, okay. So CL1 is going to be responsible for red fluorescence, CL2 is responsible for infrared fluorescence, and DD is going to be something very similar to a forward scatter in a flow cytometer. If you have done a flow cytometer experiment. So forward scatter is going to determine the size of the cells, right? So DD is something that is going to determine the percentage of aggregation of the beads, whereas these are magnetic beads and these are known to stick together. So you have to ignore those readings that come from clustered beads, ideally. So that is the purpose of a DD channel here. DD doublet discriminator, avalanche photodiodes here. Right? So we have a hydrodynamic focusing system in place that is very similar to a flow cytometer, wherein the sheath is going to be injected into the cuvette at a particular speed, versus the bead is going to be injected into the cuvette at a different speed. Okay, and the injection of sheath is going to form a core around the bead, which is going to be lined up in a single line, which will help in interrogation of both the lasers at the given point of time. Okay, so this is how the doublet discrimination gating occurs. So you can see, if you can observe two different red lines in the graph. So one is going to determine the lowest possible size of, or lowest possible fluorescence that is going to come out of the bead, and the larger and the other red line on the further to the right is going to determine the highest possible fluorescence that is going to come out of the bead. Okay, whereby gating the fluorescence, you are going to gate the presence or absence of aggregated beads or shattered beads.
Harish,
Right.
if you can finish, we
Sure,
are
sir.
almost closer to the allotted time.
2 more slides.
Okay, great, please.
So why it is ELISA versus multiplexing? So increase the amount of information, reduce the amount of error that is present, decrease the sample volume requirements, yeah, especially in case of plasma and serum from a patient who is undergoing therapy, plasma serum from a mouse molecule, so which from a mouse model, which limits the amount of plasma and serum available to you to experiment. So thereby increasing the data that comes out of your experiment by having the same sample volume. So this is the power of the technology, wherein if you are working with 48 different cytokines, okay, and the number of samples is 80, you would need to have you would need to invest around 87 hours of work and around 2.5 ml of sample in an ELISA experiment, whereas you would need just 12.5 microliter of sample and lesser than 4 hours to collect the same data points from the multiplexing technology as such. So these are all the available markers with us in form of kits for this technology. And we have the software to support the instrument. We have reagents to prepare or manufacture your own kits, wherein you can have 2 antibodies or an antibody pair, and you can couple them by yourself to the beads, and then you can have your own assay developed. So in a nutshell, we have all the things that can help you in getting your analytes measured from a small amount of sample with larger data point, ideally. Okay, and apart from good service and support, we do have, okay, so this last part is not related to the technology. So this is something, since this is a virology institute, we would like to introduce this to the audience here. So for any sample that requires, that is very difficult to amplify your gene of interest from a very small amount of sample. So this technology is going to be helpful for you. We call this droplet digital PCR, whereas QX200 is a 2-color instrument, and we recently launched, it was launched yesterday in Bangalore. So we have a 6-color instrument as of now. If you are interested about this technology, please do visit us in the stall, and we'll be happy to explain it to you. Thank you for your time and support. And if you have any questions, I'm ready to answer it. Thank you.
Thank you, Dr. Harish, for the excellent presentation. since we do not have a, it's already late. Yeah, if there is a pressing question, definitely, please go ahead, madam.
I have a question just for my knowledge. You said that we can, from a one well, we can even go up to 48 different sample reading.
Yes, ma'am. So what we have as a kit is that?
Actually, there are lots of kits available in the market. Like I know about the thermo, where you can go up to 36 plexi.
Yes.
So, just my question is that when these are ELISA plate based reader, so how you are saying that from a one well you mention about the two different color. I'm asking a question from the point of view of understanding this. When you are having a 36 parameters coming from a single well, how the reading discriminate between the 36 parameters?
Right. So, as I said, as I was talking about the dyes, okay, which are coated on the beads. So, let's say I have bead 1 to bead 36, okay. I take 2% of red dye and 98% of infrared dye coated to bead region 1. I take 5% of red dye, 95% of infrared dye coated to bead region 2. So these two bead regions will have its own spectral signature because of the percentage of dye that is coated on the beads are different. Like this, I have 36 different permutation and combination coated for 36 parameters.
But all those reading from a single well?
Yes, yes.
Reading?
Yes. Yes, yes, ma'am. Definitely, we can connect later.
Thank you, Harish.
Thank you.
Now, anyway, I also have a couple of questions. Probably,
Sure.
Anyway, you are here.
Sure.
We will interact. Please, he is going to be there in the stall for one more day, so please feel to contact with him and discuss with him. Thank you.
Sure, sir. Thank you.
Now we are concluding this section.
Thank you for the talk, sir. Now I kindly request Dr. T.R. Santosh Kumar, sir, to hand over a token of appreciation to Dr. Mohanan Valyavitil. Thank you, sir. Now I kindly request Professor Deepak Nair to hand over a token of appreciation to Dr. Rajesh Kumar. Thank you, sir. Now I kindly request Dr. Binod Kumar, sir, to hand over the token of appreciation to Dr. Hari Srinivasan. Thank you, sir. I request Dr. Sri Kumar to hand over a token of appreciation to Dr. T.R. Santosh Kumar. Thank you, sir. Now I request Dr. Rajesh Kumar to hand over a token of appreciation to Dr. Binod Kumar.
So we are officially concluding the session. And before we go for our tea break, we would like to request the students and the faculties to visit the industrial partners stalls. And we are thankfully acknowledging their contributions. We are thanking to Eppendorf, BD, Biorad, Cellgenics, Beckman Coulter, Crescent Lab Equipments, Arvind Herbal Labs, Thermo Fisher, 50 Plus Scientific, Sartorius, Biotech India, Biolinks, Laboratory Equipment Stores, Art Lab, Promega, Biogene, Merck, Zelli Biotech, Canara Bank and Trivandrum Surgicals. We are requesting all the students, they have some gifts for you in the stall, so please do visit. So thank you. You can break for tea now and we'll meet after 15 minutes. Thank you. The tea for the students are arranged in the first floor.
is going to work and what are the basic steps. So we will go through all the basic steps of diagnostic. So my overall talk will cover innovation for immunodiagnostic product, steps, and we will talk about target product profile. We will also talk about diagnostic timelines, and we will discuss for validation how diagnostic product is validated, and then we will see how it can be replicated into viruses, like with example. So let's talk about innovation. So what is innovation? Innovation is the creation or development and implementation of a new product, process or service with the aim of improving efficiency, effectiveness or competitive advantages. So this is a definition of innovation, and let's start with different type of innovation. And here we can see on y-axis there is a market, x-axis there is a technology, and you can see incremental. Incremental says it's kind of existing reagents which can be used, like existing antibodies which can be tweaked little bit and used for like creating new innovation. This is called incremental. You can just mutate some of the residues of antibodies or hypervariable region of the antibodies and you can use. Second is architectural. When you're designing some diagnostic, you are doing some like ELISA, and if you convert it into LFA, that means you are doing architectural innovation. Same reagent, but using in a different platform. Radical innovation, it's totally different. You can see new as well as here it's also new, so all new thing. Radical is totally new. And disruptive is like new thing which actually established itself in parallel to what was existing. Fine? So when you're doing innovation, you should have a thing in your mind, what to invent, what should we innovate, how to decide our innovation. So with these, you start your innovation, product innovation, and what you do, you need to identify your like research question, focus area, need filtering, need screening, you need to invent. When you are doing any kind of like diagnostic development, you need to just generate a concept, you need to select the concept, and finally you need to implement and integrate all those things into like when you're doing innovation. So what is needed to be focused? So basically for diagnostic, we need to focus on diagnostic or product innovation. We need to focus design, develop and deploy. Before earlier session has shown lots of thing on discovery. Professor Devinder Saigal has shown discovery as well as implication and whatever it is, but that was in therapeutic. But when you go for product innovation in diagnostic, you need to follow 3D. Discovery you can do for your own sake, but major focus should be design, develop and deploy. And you need to choose a rapid diagnostic, like it should be point of care, because with sophisticated instrument, it will be not like field deployable, and it would not be like easy to do with everyone. So like if it is done under the tree or in another, like beyond the hospitals, it can be done at home. So that diagnostic is going to give you some idea. So for that, you need to think how to do like innovation and how it will impact. It will give you the early diagnosis of infection, and it can also help in treatment, deciding the treatment regime, and later on the outcome will be like you can have a better cure once you diagnose early, and it will actually maximize the effect. So first step is to filter the need. So you can see in the left panel all the blue balls. These are needs. You need to first filter them. What are the specific needs while you are developing your diagnostic or you are doing a product innovation? Some needs will be filtered. Then you will generate a concept, how to develop a diagnostic, and based on that concept, you will generate a final concept and you can have your model. And for developing all those things, you need to enlist all the requirement of your product, the final product. So you need to have a good profile of like target product if you want to develop. So this is known as target product profile, and that considers everything starting from unmet need and starting from the inception of project. It will also include clinical validity, human factors, regulatory requirements, how you will like channelize your regulatory requirement, how you will go for like a specific problem, like how will you analyze your data, how will it reach to the end user, how it will go to the market. So you need to consider all those things, and that should be enlisted in the target product profile when you are developing a diagnostic product, fine? And not but the least, you should have a prototype. Without prototype, you can't proceed, fine? So prototype is like a picture on a social media which gives you 100 likes or say 150 likes. So prototype is basically a picture of your product which will give you so many likes. If your product is like user friendly, you will get good appreciation for that. So when you're designing a prototype, every step requires review when you are designing a prototype, and you need to do refinement, and it goes in a iterative manner. So if post refinement the prototype is not working, you again change, come to the square one, like 0, and you again start designing your prototype. Again you put it into review, and then again you refine, and finally you decide that okay, fine, prototype has been designed. So coming to prototype, what is prototype? Prototype can be anything. Any of these categories can follow in the prototype. Maybe sketches, diagrams, or graphics, or spaces, videos. So how do you do design process? You first understand the problem. What is like problem with the health? And then you observe it, and then you like visualize, and then you develop a prototype, and finally this prototype is implemented, and again you can see it goes here. So there are 3 steps basically for prototype development. You get inspired first. Okay, fine, I need to develop this. I'm inspired. Okay, then you evolve your thoughts, and then you go for validation post developing prototype. So what happens if it is fine? Okay, your prototype is being done. If it fails, you again reach to 0. So what are the uses of prototype? Actually, it gives you idea where you are going to fail. Fine? So you have at least a blueprint for your product. So like you can see here, here is cost at y-axis, and here is like project timeline, and you have seen you're developing like prototype, and you failed here. Say you failed here. You saved lots of money. Fine? So that is required for like project implementation. If you're wrong, if your prototype is wrong, so better you fail early. That's why prototype is developed. Fine? So, and prototype is also required for giving a proof of concept. So, let's use all these information to proceed for like product innovation. So, before I start, I would discuss other steps also, like before you are designing any product, you first choose what should be the biomarker, what should be the biomolecule, which we are going to use as a diagnostic intermediate. So omics is very famous these days. From omics you can have a biomarker, and you can develop an antibody against that. So now we will talk about tools, and since this conference is totally based on antibodies or maybe binders, so we will focus only monoclonal antibodies, and you can see for invention and innovation, tools for health product has diagnostic intermediates or binder maps, so which is being developed by two technology. Majorly two technology, which has been discussed earlier. You know, one is hybridoma and another one is fast display, and here you can see hybridoma is natural system, fast display synthetic system. So you basically engineer them to get a full length antibody, or maybe you can use other format of antibody for your diagnostic purpose. And next one is like post having diagnostic intermediate, you will think of designing an assay. So I am giving you an example of like lateral flow assay or immunochromatographic assay. So basically, it's like similar to ELISA. ELISA can be miniaturized into this. It's based on capillary action on the membrane, and it's an immunochromatography. So basically, you need to have a different like intermediates, and there are several steps. Say, if you are using your antibody as like intermediate, so it should be coupled with which kind of nanoparticles that should be seen, and what kind of antibody we are going to use in like lateral flow assay, whether it should be good or like having a strong affinity or low affinity. That should also be decided before you are doing like assay development. You need to think about other materials of LFA, like membrane, conjugate, sample pad, wick pad, test strip also, and other things like how longer it will take in like running the sample for like, say 1 centimeter or 2 centimeter. And you also need to have like data analysis, like how will you analyze the data based on what all reagent you are going to use in your product. So another thing is clinical evaluation or clinical validation. We need to like see. So in diagnostic pipeline, it holds very promising position. You need to have a good clinical evaluation, and this is like requisite, like it should be reaching from bench to bedside and to product in an iterative manner. Suppose that your product fails, if it recognizes erroneously, suppose that there is a disease, but it's showing a negative result, so it will again come to the bench and then again to the bedside and to the product. So it will be like in a translational research, this cycle goes on in an iterative manner. And we will not get into the definition of these biostatistical terms, but in diagnostic, sensitivity and specificity plays major role, and other two are PPV and NPV. So we will just see pictorially. See, here it's like health, and here it's like disease, and here is the cutoff. So it helps us to decide cutoff, where we should start recording the data, whether this is positive or this is negative. So These are the areas which are going to give you true negative, true positive, false negative, and false positive. You can see by arrow. And in a 2:2 ratio, sensitivity gives this formula. Yeah, I'm just coming to that. And specificity is also like shown by 2:2. And this is a overall like diagnostic timeline. You start with point A, like, and point A and B comes into research and development. Point C is in evaluation, and here you develop proof of concept, and D and E comes into implementation, where you go for regulatory approval, and like you go for implementation. So based on this, I will just show the example of one antibody which was developed as a diagnostic candidate for S. typhi. So for developing a monoclonal antibody, we first chose a target, fleecy protein, which is a flagellar protein, flagellar antigen. And we did in silico analysis, and we found that there was some hypervariable region. So we identified that, and we generated recombinant, as well as we isolated native flagellin also, and we did hybridoma technology with a native flagellin, and we generated monoclonal antibody, and we got screened it with that hypervariable middle region, which actually is a specific to S. typhi. And it's so specific, like it can differ from other serovars also. So we have basically exploited the existing concept of like seroreactivity. So most of the strains are checked against that region. So we generated that monoclonal antibodies, and we converted into like assays, and we see here that it was binding to the soluble antigens of different forms of like flagellin. So there was so many forms used, and it was specifically binding to the typhi antigen, but not to the typhimurium, which is the closest serovar, which actually infects mice and causes same symptoms as typhi causes us. So it's a model for study of all the typhi related like disease changes or host pathogen interactions. So it's being used like that. So we have seen that it's not binding to the flagellin of typhimurium. We also checked on denatured antibody, result was same. Then we checked our clones on like native antigen. So we did flow cytometry over the cell surface. We use antibody for binding native flagellin over the cell surface, and we got that it was specifically binding to typhi, and there was no cross-reactivity. You can see in the circles here. And we use destabilizing agent. As you all know that like human samples has different pH, or you can get samples in different conditions. So the antibody should be like so robust, it can actually, for diagnostic, antibody should be so robust that it should not get affected with a change of condition. So we screened under extreme pH condition, as well as antibody antigen interaction destabilizing reagent, and we found that these were very robust antibody, and these were binding at even pH 4. And finally we did kinetic analysis, and we found that there were two categories, robust binders as well as moderate binders. So this was used for kind of proof of concept generation in an assay, and it showed that it was binding like 15 nanogram per ml concentration in like at lower concentration, and we gave the prototype. Finally, it fetched THSTI patent, as well as publication, as well as technology licensing. So this was kind of prototype which was given for like product development. Know-how was transferred. now we will talk about how this information can be replicated in virus, since this is a virology like institute. So we'll have, we'll discuss in 2 slides more about how we can target viruses. So you can see with this slide, we can see here that there are different potential areas where you can design diagnosis, and see viral antigen detection is one of the area, and you can have a immunodiagnostic assay by detecting antibodies, maybe IgM, IgG, IgM for like early infection, IgG for convalescent. So here you can see all the graph is shown. So with this graph, we can conclude that this is the window where you can target. Why? I'm coming to the next slide. And because here you can specifically detect the viral antigen, and this will actually give the idea. So currently, whatever like kits are available, commercial kits are available. So it's a comparison for the same. So some of them are isolating virus, some of them are detecting virus post-isolation, and NAT is also there. And there is one NS1 detection kit, which is based on immunodiagnostic, and antibody detection kits are also there. So you can see these are the matter, like here you get more accessibility, like you will get more antibody, you will get more antigen, this side if you go, but the confidence will get down. So here you can target your like product development or diagnostic development, where everything is like in equilibrium, and this is also superior because it will give you better specificity, and it will rule out the cross-reactivity with other flaviviruses, like some of the antibodies which are generated naturally generally react with other flaviviruses coat antigen. So you can target it like this. You can think about the specific antigen, and then develop your monoclonal antibody for doing all like product innovation. And like, is there any scope? There are competition also for, if you are developing some like product, there are always a competition. So see, there are certain commercial, I have shown here only make in India products, like these are the diagnostic product which are available in India, made by an Indian company. So yeah, there is scope, because this is dengue day 1 diagnostic kit, which it detects dengue at day 1. But it detects all the NS, like NS1, NS2, NS3, NS4, all the antigen. So basically all the strains are being detected. So this is, and it has not been approved yet by FDA for full dengue diagnostic. And another thing is chikungunya diagnostic kit is available, but it only detects IgM. So there is no antigen detection kit yet. And the most of the reagents in different diagnostics are used are from like exported the clones and the antigen, and then they assemble all these things. So most of the antigens are like from a different origin. They are not from your geographical region. So there is a chance of like, there is always a potential of developing your own reagent and your own diagnostic. So there is certainly a need for diagnostic product development. So with this, I would end my, if you have any queries, you can just like mail me on these mails. Thank you. Any question? Okay.
Questions from audience?
this is regarding the need in the country to import, some raw material which may be for many of these kits that we manufacture in the country. Some of the components come from, other places outside India. So, how acute is this issue? In your thoughts and comments. I mean.
So basically, you are asking that suppose that the reagents are coming from abroad or maybe imported from somewhere, and how acute is this issue?
Yeah, I mean, supposing I have to make a kit, and I realize that this component is not made. We don't have that raw material in India, so it comes from whatever ABCD country. So
yes.
You are not able to assemble your kit because the raw material is there.
Exactly.
The second thing is that you might have a pandemic like COVID recently, and we just ran dry of this thing. So how acute is this problem, and what we could do as a country to solve this?
So basically, most of the kit which are being used in India are exported from outside, and they use reagent from outside, and the reagent or like the variants which are prevalent there have variability in their sequences, and like they are not matching to like Indian. Suppose that if you talk about like typhoid only, so we are in the endemic region. But if somebody in like UK is developing antibody against typhoid or maybe diagnostic against typhoid, he will simply take the strain which is available there. But we need to have a local strain, clinical isolate for generating antibodies. At least we will have an idea. It has evolved through our like disease manifestation clinically. So we need to consider those variants, and then we need to develop the reagent for like implication in our own population. And it will be basically targeting maximum to our population. So that goes same for therapeutic also. You need to have a clinical isolate, and you start targeting that. So before designing diagnostic also, you need to have like upstream this thing.
Do you have any question? Thank you, Dr. Chandresh.
Thank you.
So this brings us to the next speaker. Okay, so the next speaker is Dr. Chitra Arvind, and she is going to be talking to us about how to protect intellectual property rights for monoclonal antibodies. And this is a very important area for all of us, and I'm sure you will benefit hearing her.
Dr. Chitra Arvind is a registered patent agent and an advocate enrolled in Delhi Bar. She has been practicing in the field of intellectual property rights for about 2 decades. Her expertise lies in discussing with inventors to identify patentable subject matter, drafting, filing, and prosecuting patent applications within and outside India. She has to her credit the grant of hundreds of patent applications and success in several contentious matters. She is a visiting professor at several academic institutes, delivers lectures, conducts workshops at various forums, and is a member of many professional organizations. We welcome you, ma'am.
Thank you. Thank you for the kind welcome, and I believe I'm the last speaker of the day, and I'm the person standing between you and going home. Sorry. I'm going to take 30 minutes of your valuable time. So far, all of us have been discussing science, and science is black and white. All lawyers, we wear black and white, but law is not black and white. Law is gray. Yeah. Hello? Yeah. And law is gray. So I thank Dr. Rajesh for keeping my talk as the last talk of the day, because we all want you to think when you go home and come back with questions tomorrow in the workshop. And this is what I want all of you people to do, because my presentation is not going to give you any answers, but I hope that it will generate many more questions in your mind. So what is patent? All of you here is filled with Saraswati. But just because we have Saraswati, don't we not dare to have Lakshmi? Why? Patent is one of the way where Saraswati we catch by hand and take her to walk to meet Lakshmi. Okay? And this is what we are going to do today, and see how to take Saraswati to meet Lakshmi. But why antibodies? Why are they so special? Our chemical molecules are in the size of a pinhole. But look at our biological molecules, size of a football. But do you think how we deal with chemicals is the way that we should deal with biologicals? Is it how patents are to be dealt with? Unfortunately, yes. Because the law doesn't differentiate between a pinpoint and a football. And whether it is a biological or a chemical, you have to undergo all the basic tenets of patentability, which will be, first of all, to understand as to what is your invention, what is the unique and distinguishing feature of your invention, do you need to have a patent, and what you need to put inside the patent application so that it becomes a very well granted patent, and how to structure the claims. So, every patent is determined for three basic things. What is novelty? Novelty means it has to be new for the first time. It has to be inventive, which means it should be unobvious, or it should be beyond what a normal person would think, and it should be useful. Okay? Unfortunately, that alone is not sufficient. It should not fall into a non-patentable subject matter. And if it falls into a non-patentable subject matter, it must have few exceptions built into the specification. And you have to provide data for it. How many of you have put a poster here today and tomorrow which you have not patented? That is disclosure. That is novelty. Dr. Rajesh is going to print all of them into a conference proceedings. Okay? So, if you go out and tell your invention to somebody, so all of you are, I don't know, I'm not asking anybody, but you are all going to go home to your room today over a cup of beer or a cup of coffee. You are going to tell your friend, this is my invention. Your friend will go and publish it. How many conferences it happens? There, you lost your novelty, you lost your patent application. Secrecy is the key to patent application. Anything that you say must first go into a patent application, which includes all your oral talks, all your posters. So how do you do that then? So you do not disclose the key ingredient. You say it is XXX, you say it's YYY, or you say 1, 2, 3. I want you children to pull out your posters today, hide your invention. Tomorrow there is a turbo talk. Okay, people should not be able to go and repeat your turbo talk. This is the essence of patenting. Okay, so there cannot be inherency. Inherency means what exists, exists. Okay, you cannot rediscover that and say it's novelty. So since this talk today is about antibodies, so what is novelty in antibodies? When you talk about novelty in antibodies, when the nature made something in an antibody, like Dr. Rajesh was talking as to how you can isolate antibodies from humans, do you think you deserve a patent for it? Mostly students, so I'm daring to ask you all questions. Do you think you deserve a patent for it? Yes? Why? Nature made the antibody. What did you do? Unfortunately, our judges don't think so. They say that if it is exactly found in nature, since we cannot find Mother Nature to give the patent, the law doesn't give anybody a patent. Okay, so if it is found exactly in that of nature, like in case of antibodies, if your mouse has made it, if your humans have made it, you cannot get a patent because it is considered as anticipated and nature made. Okay, so how do you do that then? You have to change it, and that becomes your human intervention. Obviousness is a little more difficult than novelty. In case of novelty, we just take one and match it exactly with anything that is already existing. So there you are not going to get a patent, but you take something and you modify it a little bit. So you take an antibody, you have made it a chimeric antibody, you have made it a humanized antibody. Full human antibodies? No. Full mouse antibodies? No. But you have chimerized or you have humanized. Okay? Is it good? You get it? Yes, you do. But then, even in that change, they will ask, is this change routine? Is this change known? Okay, in an exendin, okay, Lispro insulin, okay? It's an insulin where the lysine and proline were interchanged. Got a patent application, okay? But if you replace serine with threonine, or you have other like amino acids with like amino acids, such modifications are considered to be obvious, and it is not possible for us to get a patent because they will squarely fall within the second test of patency, which is obviousness. So how do you get out of the obviousness in case of antibodies? You have to show an advantage. So you made a change, the change is different. It is even though for the same target, even though produced by the same method, you have to show that there was no reasonable success. How many of us record negative experiments in our lab notebook? Thank you. And that is what you want in a patent application. Dr. Rajesh spent 4 years of his PhD, he said, and he did not get something. Okay? But you think that 4 years worth of lab notebook he has? He has. Okay. Who said this? That, there were 1,001 ways of a light bulb not working. Okay? This is what we look for in a people. When you do research, We know that research is not easy, but you have to tell. Okay, when you tell that there is a cross-reactivity, there is an increased selectivity, there is an increased affinity, or a new or improved function, there you clear the test of obviousness. The last test is very easy, utility. Does it have any industrial application? Yes, it does. Okay, that's it. So very 3 simple tests. All you have to do is to cross the bar of novelty, cross the bar of inventive step, and that of utility. But is that sufficient? No. We have all been defining antibodies scientifically. Let me define it patent-wise. How do you define it patent-wise? So either you take the antibody and define it by the antigen to which it binds. So when you talk about the antigens, there are linear epitopes, there are non-linear epitopes, okay? Or you talk about the structure, like this is the sequence, nucleotide sequence, amino acid sequence. Or you talk about the function, or you talk about combination, antibody A plus antibody B, or antibody A plus a drug B, or a composition, an antibody present as a liposome. That is a composition. Or you talk about the process in which it is produced. Kohler and Milstein, their name we have heard again and again. They won a Nobel Prize, but they never filed for a patent. But can we file for a patent today? No. Well, Kohler and Milstein got their funding. We need our funding, so let's better file our patent applications. Okay. But today, if you go with a hybridoma for a patent, you will not get it. Okay. What other technologies have become routine? Many others. Okay. So if you are going to define it by technology, it is very difficult. And of course, there is a method of treatment. So I am just taking this quickly through how to patent when you have a new antibody at hand. Supposing your antigen is new, okay? You are the first time coming with that of the antigen, okay? But you have not given the full sequence of the antigen, and you are defining the antibody to go and stick with the antigen, but the sequence of antigen is not given. Sorry, not patentable, okay? They will say there may be many antibodies which can bind to that particular antigen. And therefore, you will not be able to get a patent. You have to show that the epitope is new, the antibody is new. Okay? But you have clearly defined your antigen. Okay? You are fully sequenced. Then anything which binds to the new antigen, anything, any antibody, discovered, not discovered, the whole antigen is yours. Small molecules are yours. Any binder, if you come up with the antigen for the first time. Okay? Imagine the extent of protection that you will get if your antigen is new. Okay? When earlier, when antibodies, it was possible to define with some antigens. Now it is not so. They ask whether the antigen is known and does it form a part of the already known family of proteins. If that be the case, they will say, okay, for example, GPCR proteins are already known. You have found a new family of GPCR proteins. They will say, already antibodies for GPCR proteins are known. So even though your protein is new, you are not going to get anything if it is from a known family. So the chances are very less. So you have to show the antigen is completely unknown. You have to show that all epitopes of the antigen you are showing for the first time. It is a difficult task, but you have to show. Not only that, you have to show where it is binding, how it is binding, and all these tests has to be shown. And again, it gets interpreted differently in US, India, and EP. We attorneys are very bad. We tell When you file in India and get a patent in India, we will check for novelty inventor step across the globe. But if to get a patent, you will get only in India or US. So the case law for US is different from case law in Europe and case law in India. So what you can get in India, not necessarily you will get in US, not necessarily you will get in Europe. Okay, to have one specification to satisfy all jurisdictions is a challenging task. Okay, so who is this? This is Amgen and Sanofi. Okay, in the first slide we saw, all major antibody manufacturers. Amgen we all know, Sanofi we all know. They both fought with each other. Okay, Amgen had one whole antigen. Okay, in the written description they have not described the entire antigen. So what did our people say? The people, Federal Circuit is US. Federal Circuit says, you have not defined the entire antigen. How do you think that I will know what antibody is going to go and sit on it? You are all scientists, I am not. Tell me, is it possible for you to go and sit? Tell what antibodies are going to sit in that antigen. So US said, it is not sufficient for you to claim the entire set of antibodies over that of the antigen. But Europe, what they went is, they took it to appeal. And they amended, Amgen got smart because they have already lost in US. They have amended the claim so that they were able to give the full sequence of the epitope. So Amgen got the entire epitope. And any antibody, tomorrow you touch the epitope, you will not get a single patent because the antigen and the epitope is protected by Amgen. Okay, so let us be the person who invents the antigen. This is again similar, and every place where you try for such an antigen or an epitope claim, you have to define the entire epitope. How you will satisfy? There are few more examples. In every place, you have to show that the epitope is fully defined. And nowadays, people are not satisfied with a single protein or nucleotide sequence. If you give a nucleotide sequence, there may be many proteins which are possible out of that nucleotide sequence. So people are not satisfied with nucleotide. You have to give protein, and not only one protein sequence, all 6 CDRs has to be defined if you have to get an epitope claim. So defining the antibodies by antigen is not a good idea. So you define it by structure. So if you are going to define it by structure, all 6 CDRs minimum, okay, otherwise no chance. So in US, if you define it by structure, it is very easy to get a claim. And in EP, it's unfortunately rejected because they say structural elucidation is obvious. You should ask it to a PhD student who spends 5 years, elucidating the structure. But unfortunately, our EPU is not in sync with me, and they are saying that structural elucidation by sequencing is very obvious and does not involve inventor step because it is through known techniques. I'm sorry. So what do you do? But then, what do you do? There is a function. So it's very difficult to define by structure. It is very difficult to define it by antigen. So you define it by function. So you say what is the neutralizing activity, and then you have to show how the neutralizing activity is different for this particular structure, and how this neutralizing activity is better. Okay, so what is the best way? The best way is to use a combination of all 3, because as a lawyer, when you discuss with a judge, you first throw one arrow. That doesn't go and sit. Then you throw the other. That does also go and sit. Then you throw the third. At least one of it will sit and you will get the patent. And it is not possible for anybody to add the information after a patent application is filed. So in the beginning, we ask for antigen sequence. In the beginning, we ask for antibody. In the beginning, we ask for all negative experimentation, saying that it is not a matter of routine. It was so difficult. It took a whole set of lab of people for so many years to come up with this particular antibody, and we give a function. So you have to put all your arrows well in advance. Combinations and compositions are more easy. Combination itself means it is a matter of two active ingredients. If two active ingredients are combined for the first time, then it becomes very easy for you to get a patent. Composition means, say for example, you take trastuzumab and put it inside a liposome or a gold nanoparticle. There also it is easy, but the novelty, inventive step and utility should vest within that of the composition. Hybridoma technology, I know it's used very frequently, but it has been used so frequently now that it has become a matter of routine, and it is very difficult to get, a patent application on this. How about method or treatment of use? It is not granted in many countries. New use of known substances or repurposing or method of treatment is not grantable in many countries, including India, but US and Europe still consider method of treatment. So if you have a known antibody with a known antigen, okay, and there is no combination, there is no composition, and the functionality is same, but the use is different, then we try to use the method of treatment, but only after we fail in the first few tenets. This happened for Herceptin, trastuzumab. In trastuzumab, people tried, to have a new method of use, but it was rejected by the court because they said that, it was very well obvious that wherever Herceptin is not forming a treatment, it is very difficult. So the big question in antibodies is it a matter of routine experimentation or is it not? Or are we placing undue burden on the people to ask for so much of work? So how do you have to overcome? You always have to overcome by using a combination of claims. Give the function, give the structure, okay? It should show failed experiments. It should show lack of inherency. If you pick up any antibody patent application, it is not less than 100 or 200 pages, okay? Unless you give all this, you will not have enough ammunition. You have to have all written requirements, okay? If you are saying CDR, it is not sufficient to give nucleotide sequence, you have to give protein sequence. Nowadays, EPO is also asking for X-ray crystallography structure if it is conformational. Okay, all this is required, but it is not impossible. So it is still possible for us to get all of them. So you have to use a combination of all. So what is this? Intellectual property rights cannot only be defined as patents. Okay, it also includes non-scientific areas such as trade secret, geographical indication not so much in India, or in this you have patents, you have copyrights, you have trademarks, which it protects the name of any particular generic substance that you give, or copyright which protects the written literature. Trade secret is like when you do a lab, you know that there are 1 or 2 experiments which will only work. Say for example, when you use collagen methanol and not sigma methanol, but that's a trade secret that you hold, okay? All these combined together is intellectual property rights. And unfortunately in India, we have a Biological Diversity Act or an NBA. So anything that you take from humans or you take from any samples has to be protected or applied for under the NBA, otherwise you don't get your patent to be granted. So these are things that you should note. So people will be wondering, why is it so that I am talking about trademarks and trade secrets? It's very important. Herceptin, it was launched Roche does not have a patent for Herceptin or trastuzumab in India. Okay? Herceptin, which was there, is the brand name or the trade name. Okay? Two of our Indian generics, Mylan and Biocon, launched this product and they had injuncted by Roche. Not because they infringed patent, because Roche did not have a patent. Okay, in their product brochures and literature, they said we are launching a biosimilar to Herceptin. Okay, it is a huge mistake because Herceptin was the trademark or the trade name of Roche. So they sued them under Trademarks Act. And while making the product brochure, copy paste of paragraphs, any 10 words lifted in conjunction is a copyright violation, and therefore Roche was effectively able to stop them from using or launching the biosimilar of trastuzumab in India. So it is not only patents that you have to think about, you should also think about allied IP. All of us know this mouse, the Osgene mouse. Okay? So this is also an effective way. Like you see all this Osgene, these Osgene are people who make mouse. Okay? For your in vivo testings. They say, oh, you knock a gene off, they will knock a gene off and give you. Okay? These people advertise their mouse like this. So these Osgene toys are equally popular and they have logos like this. So this is also, you know, a way of marketing your product. So don't only think about patents, you think about everything. So not to discourage all of you, it's possible to get patents for antibodies and other biotechnological substances, as we will discuss tomorrow. But we should know what to add in the specification. There are many things which includes your failed experimentation and presentation. As scientists, okay, we always tell the truth. As lawyers, unlike the popular perception, we don't lie. We just know when not to tell the truth. Okay? So, how to present your antibody inventions is an art, but if presented well, I wish each and every one of you will take the walk in the park with Saraswati to meet Lakshmi. All the best.
Thank you, Dr. Chitra, for a brilliant talk. The floor is open for questions. Okay, while students are formulating their question, you indicated that it's a better bet to patent or try patenting the antigen over the antibody. So in this age and time when, you know, almost everything is genome sequenced across, what are the chances that you will come up with an antigen which, you know, for which we don't know anything?
Sir, the chances of getting an antigen which is completely new is quite difficult. I'm in agreement with you. But if in that case, what we suggest people is don't try to patent the entire antigen. Try to look for epitopes and show that these epitopes were not identified as the most critical epitopes in the entire antigen. This is known as a selection patent.
A second question would be that the requirements with regard to the structure as part of the application, you were indicating, you know, to increase your chances that, you know, the antibody is patented, you know, having a structure with the antigen would be of help. Again, in these times when you can have, you know, programs like, you know, AlphaFold 2 and others which can predict the structure by just in silico. So what are your thoughts how this would work or would not work?
It's not working anymore, sir. That's why we are recommending that, you know, a simple structure is not sufficient. It's better to combine, you know, an epitope plus structure plus function. Supposing, you know, structure, epitope is not novel, but your antibody, let us assume, though the chances are very slim because, you know, it will be only a complementary protein. Let us assume your antibody is novel. Or let us assume that you have taken the antibody and made it in a recombinant manner such that its binding affinity is increased, or its viscosity is reduced, or its functionality is more, or its half-life has increased, or instead of 1 dose a day, you will be able to give 1 dose a week. Those are the things that we need to highlight, and that will become the body of the patent. So every patent has to be crafted to perfection.
Questions from students? Yeah, please.
Ma'am, you said that we have to protect the main key ideas that we have, we think that which is innovative. So how we can do it in the presentations as a student?
First, as students, your presentation should be within the lab only. And every one of your slide must be marked as confidential. Okay, the only other place that you can go and freely present is to a funding agency. But even now, I work with a lot of corporate clients also, and none of them go and present their entire work like that. For example, if you have an antigen and you have sequenced the entire antigen, okay, so what you do is you put for one portion of the sequence, which is your epitope, as A, B, C, D, E, and not give your actual sequence. This is actually working in chemistry also. Say, for example, if you have a molecule or a derivative, people only give the base molecule, they don't give the substituents of the derivative. It is accepted by funding agency. On the contrary, the funding agency is more happy because they know that you know the confidentiality of the work. Okay? So between the lab with people, okay, it is always advisable to mark your lab books confidential. There has to be a lab book record, and the lab book record has to be maintained by your PI. These are, practices that we should follow as good laboratory practices. Thank you, sir.
So we have come to the conclusion of our first lecture day series. I hope you all had a pleasant and informative session today. So we are requesting Dr. Mohan Vallivittal to hand over a token of appreciation to Dr. Chandresh Sharma. So please. We are requesting Dr. Binod Kumar to hand over a token of appreciation to Dr. Chitra Arvind. Sir, please. Now we are requesting Dr. Rajesh Kumar to hand over our token of appreciation to Dr. Devendra Segal for chairing the session. Is it fine? Sir, Mohan sir, can you please receive? Yeah, okay then. So that marks the end of our formal session for the first day. So we have some quick announcements to make for tomorrow. We have already announced you that we will be having a briefing for tomorrow's session. So all are requested to arrive by 8:15 sharp, because we will have a briefing for the hands-on module by 8:30, and we'll start the laboratory work by 9 o'clock sharp. Since we are having time constraints, we have kept each module to 1 hour 45 minutes to 2 hours. So if you in the lab at the right time, you will be able to finish it, and you will be able to attend all the 3 modules. And there are 12 modules where each of you will receive 3 modules, including 1 IPR session, and it will be 2 hours duration. And the instructions for maintaining biosafety in the lab has already been given to you. And we have coordinators for each group. We have mentors for each group. So if you have any queries or worries during the hands-on training module or while arriving at here, you can please contact our help desk or the coordinators. So I think that is for the briefing for today, and tomorrow when you arrive by 8:15, we will start the briefing by 8:30, where you will be guided to your respective labs. For the first module, you will have your mentors and coordinators with you. If you have any queries, please feel free. So to officially conclude the first day, on behalf of the team IAV, we are thanking all the speakers and the chairs to be here and then able to present their work and holding the whole session on time, and they have helped us to close our session on time. Even though we ran a little late by 30 minutes, we were doing fine, I guess. So we were having a really informative session. We are very happy to host an amazing crowd like you. You were very active, and we are expecting the same active participation from your side for tomorrow also, because we are having surprise gift from mentors for you all. So kindly read about your module and please be here on time. We will see you all tomorrow morning. Thank you.
You see, I was talking to one of my friends and asked him,